Anem seient i començarem d'aquí un minut aquesta mena de trobada tan festiva. Hola, Pere. Turista. Okay, I think we can start. Però no se sent? No vol dir... Okay, pay a little bit of attention to me, please. Okay, very good. Okay, so we start. I think it's time. There is a very long session ahead of us, so I think it's good to start a little bit on time. First of all, welcome everybody who is here, everybody who may be connected. We are very happy to organize this session. I'm very glad to be here with so many people, so many young people, and so many people of my age. So thank you everybody for being here. I would say already a couple of years ago, we were talking with Pep and realizing that time is going on and Carlos was going to be retired soon. So we thought it was very nice to organize a session to celebrate with him scientific achievements. And then we asked him, do you want to do this session when you are retired, or can we do it now? And he said, no, now, do it now. So this is why this is now. Carlos is not retired, he's emeritus. I don't know when he's going to be retired, but but he's still active. So this is not a session to say goodbye, but it's a session to celebrate aquatic microbial ecology. So, and this topic of aquatic microbial ecology, I would say that it was born in the last century, of course. There are like two seminal papers, one in 1974 by Pomeroy, where he put bacteria inside trophic webs, so bacteria were a trophic component. And perhaps another seminal paper was in 1983 by Farooq Azam, where he's proposed the concept of the microbial loop, so the incorporation of dissolved organic matter to classical food webs. So, and there has been a very long survey, a journey since then, since the 70s and the 80s. And I think people working in microbial ecology during this time, we have been privileged because we have been able to make natural history in current times and to address very, very simple questions that were unanswered and unaddressed. And the questions such as how many bacteria, how many microbes there are in the environment, what are they doing, and who they are. So it's fantastic, and as I said, a privilege to be able to answer these very simple questions. Because we had new tools to answer these questions. That was microscopy, radiotracer experiments, and later all the molecular revolution. So during this journey of aquatic microbial ecology, Carlos has been there in many different aspects. So he defended the PhD in '81 with one of the fathers also of this field, which was Thomas Brock. He's famous by his book of Biology of Microorganisms. I have the 5th edition. I like it a lot, and that was from, I think, '88. But I think the first edition was in the '70s. And I check now in the internet, I think there is the 14th edition in 2014, and perhaps there is a new edition. So this is a book that has accompanied many generations of biologists, microbiologists, and microbial ecologists. So he did the PhD with him. He came back to Barcelona, to the Autonomous University in '81. He incorporated at the ICM in it's not important the dates, I know, but I want to say the good dates. In '89, this is what he told me just 10 minutes ago. And then he was here, and we did a very nice team. And then he did a sabbatical in 2013, and he never returned. So he stayed in Madrid, where he's now in the Center of National Biotechnology. So Carlos has been part of this journey on aquatic microbial ecology. So, and yeah, I want to say when we think about Carlos, which, I thought, what can I say about him? So I want to say 3 different things. First is that he's a very curious man. He wants always to learn. He's eager to have more knowledge. He's like this Renaissance man that they wanted to know everything. He's questioning everybody. It's easy to talk with him because he's so much interested. And so he has touched many different things, not only science, but also art, literature. There is an exposition of books that he has written in the entrance. So he's a very multidisciplinary scientist. This is the first thing, this curiosity and this need to know more. The second thing that I would say is that it's not only knowing by yourself, but he is very aware that your knowledge has to be shared to the other people, transferred to the others. So he was very much into presenting the research that he conducted in scientific papers. So that was something that we learned from him. We have to publish what we do. If we don't publish, it's not done, basically. But also, he's very famous by the speeches, for the talks that he gives. So everybody remembers his talks. And also lately, he's written a lot of books. Sometimes these books of explaining scientific things or projects or cruises. So he has also this dimension. No? And the third thing that I want to say from Carlos is that he's very generous because he has touched many different things and he has shared the knowledge with many people and he has constructed like a dynasty of sons and grandsons, which is something very recognizable. So, and that's also something why we are grateful, and this is something that we are here. And an empirical proof of this generosity is that we are here in this conference, and is that it was so easy to convince the people to make a speech today. So that was very easy. So people were very positive to participate because everybody wanted to recognize his contribution. I have to talk 10 minutes, and I think almost there. No? Not yet? And so to finish this presentation, with Pep, when we thought how we can organize this session, we thought, let's make a scientific session to recognize his contribution, to acknowledge his contributions. So let's organize a scientific session in different keywords, in different topics. Again, these different topics is an example of his multidisciplinarity and his interest, because the topics are quite broad. So the topics that we are going to listen today, and that we asked the speakers to talk, they range from starting with limnology, because he started to work in lakes already in the States, but also when he came here, he was working in Banyoles, in the region of Banyoles. Then another keyword is diversity of prokaryotes. He contributed from the beginning to this area. Another topic is diversity of eukaryotes. Those are the keywords of today. Another keyword is genomics. Another keyword, let me see, if I don't remember, is polar research. Another topic is bird watching. So he has also very interested always in all the cruises that he and his travels that has been a component of his interest. Science and art also. I mean, that was one of the first, at least here, that we started to listen his interest to combine artistic contributions and science, and finally extremophiles. So I think with that, we open the session, and we have the first speaker. So.
Sí, digues alguna cosa. Com se fa això ara? Convertir todas las herramientas. How do I do that? Ah, yeah, good.
Okay, let me listen. So the first speaker, it's a combination of Pep and Clara, and that's going to be the topic on limnology. Carlos, when he came to the Autonomous University, was working in a small lake in the Banyoles area. I think it's...
Això ho diré jo.
Okay.
Okay, thank you all for coming. We'll start with talking a little bit about lakes and limnology, and I will give a small introduction, and then Clara will show some science. And I asked her to give the talk, and she said, no, because it's too far away, Carlos, and so on. And I said, well, okay, I will give the introduction, and that's okay. And my introduction has to come to when Carlos, with the advice of Ricardo and other people, decided to go to Madison to work with Tom Brock, which is this person here. As Ramon said, you may know him because you might have one of these editions of the microbiology book, but also, and this is, I think, very important because the first book on microbial ecology was published by him in 1966. So you don't have a copy, right? We should have one. Anyway, Brock was known for the discovery of Thermus aquaticus and the growth of Thermus aquaticus in culture, and you know that the PCR comes from that organism. But he also published on things that we are very interested right now, like the production of or compounds from algae that bacteria use, organisms growing at extreme environments, and we will hear about that. Autoradiography that Carlos brought to the Institute, and several of you have done work with autoradiography. But I like this one. And this is a paper that nobody knows, but I love it. I love it because it's the study of microorganisms in situ. And he said, much of what passes for microbial ecology does not fit the definition of science. Many studies that pretend to be ecological are still using antiquated, discredited, or meaningless methods. Many studies are unfocused or do not deal with important questions. And many studies, a clear question is not formulated. I say, oh boy. And then he said, some say that microbial ecology is easy. Wrong. Microbial ecology is not easier than those other things, but harder. Because microbial ecology deals with complex systems. It requires a skill, insight, imagination, attendance to quantitative detail, even mathematical insight that far surpasses what is needed for genetics and molecular biology. So imagine yourself having to do the PhD in a place where that's the level, the discovery of Thermus aquaticus and this feeling of what is really science. So that was something. Well, anyway, this is Lake Mendota. I think the university is somewhere over this side, over here. So you probably were seeing the lake every day from the laboratory. And I would like you to get your mind in this image, because it will come later. And those are some of the papers of his PhD. And I just remember that one, the attachment of bacteria to particles. And I said, oh boy, we're still in the same place. Anyway, but there was an interesting thing on because he also did zooplankton. And he counted zooplankton and measured zooplankton, measured actually the feeding of zooplankton, Daphnia, on bacteria in an image at the time in which what we now know, which is the role of flagellates, and especially the role of viruses, was ignored. I mean, Carlos immediately, that viruses became important, came with the idea of we have to work on viruses, and there were a PhD at least working on viruses on that. So I said, remember this scale, and this is the scale of the place when he came back in which he worked. No, but it's not that scale. So we go closer. It's that thing here. And just to give you an idea, that's the same scale. This is 100 meters. That's the same scale of when you look at Lake Mendota. That's the same scale of the lake that he and some of us were working. So Carlos moved from studying Lake Mendota, the most studied lake in the world, to Lake Siso, the most studied lake in the world per unit surface area. So actually, I did the calculation in about 100 papers, six pages each. It's about 100 square meters, which is about half of the lake. So that's pretty good. So and all these famous people, if you are a limnologist, you know these names. You know Edward Birch, which is that guy over there, Chenchi Juday, well, Tom Brock, Steve Carpenter, which is very famous in lake ecology, Trina McMahon, many people that work in Lake Mendota, and these are the people that work in Lake Ciso. Some of the people that work in Lake Ciso, you recognize that guy. There's others here, also Juani, some may know Juani and Josefina, and also Monse, it's here, and Emilio, and even Klaus is here in this picture down there. So just to finish, say that that lake that you and I studied suffered some global change, some human change. So it reads, somebody working in the fields actually emptied the lake, and the lake actually collapsed because it was empty of water. So this was actually global change. However, I think that the lake has returned more or less to normality. This is when Craig Benter Institute went to sample it in 2010, and this is a picture of last year or 2 years ago, which it's not the same, but it's still same color. And going to the Lake Mendota, that's what Carlos always told us, this story of that they did using the ice of the lake, so pretending that there was the Liberty statue below the lake. But then I went and I checked that actually, since the time that he was there to now, the lake is freezing about half a month or almost a month later, and is melting about 2 or 3 weeks before that. So change is happening. Also the Daphnias that he liked so much, there was something that is called Phytotrephes, which is a very large cladoceran, came to the lake, invaded the lake, and as you can see, the cladocerans disappeared, and this thing, which is called a spiny water flea or something like that, started to eat that organism, and the whole of the ecology of the lake changed, and even the color and so on. So just to say that things change, that when we do research and maybe what we study, because we have a different perspective, the perspective that's common at a given time, we ignore things like the viruses or things like that. But then, everything that we, Carlos, every one of us has done in the research is also building up, so that knowledge that comes afterwards comes on top of that knowledge that everybody created. So lakes change, we get wider, whiter, bolder, some of us. And, but yeah, I stopped talking about limnology, and I let, about nonsense, divagations, and I let Clara talk about science.
Okay, so good morning. Thank you for being here. So I will talk a bit, I will continue talking about lakes and beyond a bit. So when Pep asked me to give this talk, at first I was like, what? I have never worked with Carlos. And indeed, even though Carlos and I spent several years together in this same institute when I was doing my PhD, I never worked with him directly. Actually, I think that our most tight interaction was during this fun trip to Seattle in 2010 for attending the ISME meeting, which was when I discovered his passion for watching birds, although I think this is the topic of another talk today. So I didn't know what to explain here today, but then I started thinking about all the things that I have done during my scientific life, and I started to notice that Carlos may have been behind many of them. So for example, as Pep said, Carlos was the one who brought microautoradiography here at the ICM. So microautoradiography is a technique that allows us to identify which microorganisms take up or use a given substrate that is labeled radioactively. So Carlos brought it here, taught it to Maria and Laura, who were doing their thesis here before me, and then they taught it to me, and I spent the next 4 years of my life in this position studying marine bacterial responses to light using microautoradiography. So thank you for that, Carlos. Another aspect that may have been Carlos's fault to some extent is the fact that he was not afraid of transcending ecosystem limits as he jumped from lakes to the sea. And this is something that Pep also did. Pep was his PhD student, then Laura, and then me, as I spent 3 years working with boreal lakes and rivers in Canada. And this is a behavior that is not so common for aquatic microbial ecologists because most people tend to focus forever on a single type of ecosystem. And it was indeed during my first water life when I applied most the concepts that Carlos had developed during his career. Because here I changed, or I moved from the microscope to the DNA sequencing techniques. And Carlos was among the first to embrace and exploit these new technologies. So he was apparently fascinated, for example, by the diversity of the least abundant microbial taxa, the so-called rare microbial biosphere, and proposed beautiful concepts such as the microbial seed bank. So as I will explain now, during my career, these are ideas that I have also explored. So this advent in the sequencing techniques truly revolutionized the field of microbial ecology because they allowed passing from the description of only the dominant taxa to the discovery that all microbial communities harbor a huge number of extremely rare and diverse taxa that hadn't been detected before, which is the so-called rare microbial biosphere, and which occupies this long tail of the taxa abundance rank curve that Carlos likes so much. So these techniques largely improve our perception of the mechanisms explaining taxonomic differences between communities, because what we thought was mostly due to the complete replacement of species between communities turned out to be, in most cases, due to changes in the abundance of taxa that were already present, but that could not be detected because they were too rare with more traditional methods. They were too rare. In parallel, the accessibility to these technologies boosted the number of microbial studies covering increasingly larger environmental and spatiotemporal gradients, and obviously this allows a much better understanding of the environmental drivers of microbial communities. However, this expansion in the taxonomic and spatial resolution also unveiled that besides environmental factors, dispersal processes also played a large role in shaping bacterial or microbial communities. And this is something that has interested me over the past years. So how does this work? Well, imagine a landscape like this. So here, local bacterioplankton assemblages will be connected by the directional flow of water, making part of the so-called microbial metacommunities. So, I don't know if you see here. Yes. So in consequence, the taxonomic structure of communities at a given point will result from the balance between the dispersal or the immigration of bacteria from upstream aquatic ecosystems and the selection of species due to local conditions. So you can imagine that trying to understand this local structure without considering this metacommunity context will lead to may lead to a very biased understanding of the mechanisms explaining bacterioplankton distribution. So I studied this type of questions during a postdoctoral stay in Canada with Professor Paul Del Giorgio, where we did several sampling campaigns trying to understand or to explore the metacommunity structure of aquatic bacterioplankton in these complex boreal landscapes. So for example, in one of these campaigns, we characterized the bacterial communities from more than 200 terrestrial and aquatic sites using sequencing of the taxonomic marker, the 16S RNA gene, and we traced the dynamics of terrestrial bacteria across the entire aquatic continuum, so from the smallest headwater streams to the largest rivers and lakes. So as you can see here, what we found was that all of these aquatic communities were strongly dominated by terrestrially derived bacteria, although their abundance seemed to decrease slightly from the smallest headwater streams located here to the largest rivers and lakes. And we found that this was due to a double mechanism. So on the one hand, we found that most of the terrestrial bacteria were progressively diluted, meaning that they were probably unable to thrive in the aquatic environment, whereas at the same time, the abundance of a few of these terrestrial taxa, which were rare in soils, increased notably towards large rivers and lakes dominating these communities. In turn, this suggested that the mechanisms explaining community assembly along this continuum might also change, with dispersal dominating in fast-flowing headwater streams and local sorting prevailing in large rivers and lakes. And we found that all this seemed to be ultimately explained by hydrological conditions, as water residence time determine the time that bacteria spend in a given system, and therefore their capacity to adapt and react to local conditions. So we could conclude that the limits of aquatic metacommunities go beyond the limits of the aquatic environment, and that dispersal from terrestrial sources explained a large fraction of the local aquatic diversity. This may not seem unexpected because we all know that terrestrial freshwater and marine ecosystems are tightly linked in nature, but the truth is that most microbial studies have focused on individual types of ecosystems, such as only streams, only lakes, or only the ocean, thus disregarding this natural connectivity between habitats. One of the implications of all this is that depending on dispersal rates and routes, different systems may carry different proportions of active versus inactive allochthonous taxa. And this is important because it will condition our capacity to interpret any links between the environment and the local community structure, because here, for example, we will have many taxa that are not actually responding to the environment. However, some of these dispersed taxa may also be rare taxa that can grow when exposed to aquatic conditions and might perhaps acting as seeds and might influence local freshwater communities. So this taxa would make part of the so-called seed bank, which is a reservoir of dormant cells that can be reactivated upon changes in conditions, such as, for example, when being transported from soils to the water. So this concept of the seed bank was developed for plants, referring to the pool of viable seeds that soils contain, but became very important in microbial ecology since Carlos proposed or suggested in this paper that the taxa or the bacterial taxa within the rare biosphere might make part of a seed bank. And to my knowledge, this is the first time that this term, microbial seed bank, was coined. So this concept implies dormancy, which is a reversible state of low metabolic activity, and hence, dormant bacteria may become increasingly rare in communities. So since then, rarity and dormancy have motivated plenty of research. However, as Carlos mentioned in this paper, not all rare bacteria within the rare bacterial biosphere may be dormant seeds comprising a seed bank, and hence, identifying the different components within the rare bacterial biosphere may help better understand the drivers and the ecological implications of rarity. So we did some work in this regard, like for example in this large boreal river, where we sampled very deeply two extreme communities. So one headwater stream and the river mouth, here. And we compared these two rare tails against 85 additional terrestrial and aquatic assemblages sampled throughout the entire river watershed. So just by doing this, we found not only that all the rare taxa within these two communities, the majority of them, could be found elsewhere in the watershed, but also that the local abundance of these rare taxa reflected to some extent their mean abundance in the landscape, comprising different proportions of terrestrial or aquatic taxa depending on their position along the river. So this means that the local pool of rare taxa in freshwaters seems to be mostly comprised by dispersed allochthonous taxa that reflect upstream history and past selection. And this is a finding that we have recurrently found in several studies. And another recurrent observation is that the rare taxa potentially acting as seeds, so comprising the seed bank, seem to comprise a very small proportion of the total pool of rare taxa. However, delineating the true size or the actual size of the seed bank is extremely complicated. For example, in this paper, Viviana and Carlos showed that DNA sequencing failed to retrieve the majority of the culture isolates obtained from a single marine sample. So this means that we may not even capable of detecting a large fraction of the taxa actually comprising the seed bank. However, the drivers of rarity may be different in marine habitats compared to freshwaters. But nonetheless, the marine microbiome is also subject to these linkages with the terrestrial environment, one of which is the so-called submarine groundwater discharge, a process in which I am currently interested because it allows me to link my freshwater and marine sites. So submarine groundwater discharge is a very relevant process in many coastal areas, as you can see in these images showing important discharge episodes in the Mediterranean. However, its ecological impacts are still poorly known. So over the past years, we have been sampling a series of coastal aquifers, trying to explore the microbial dimension associated to this submarine groundwater discharge. And among the many interesting questions related to this topic, one is whether this groundwater can also transport microbes to the sea. And it seems it does, as we have seen that increasing concentrations of this radium isotope, which is a tracer of submarine groundwater discharge, explains higher taxonomic richness in coastal marine communities, like for example in this coastal lagoon. So it seems that this groundwater actually carries microbes. However, we have seen that most of these transported taxa are rare in the lagoon. So again, this means that we need to consider dispersal and connectivity to understand local community structure and local rarity. Finally, whether this groundwater taxa could grow when arriving to the sea is unclear, but we have found that fresh groundwater from coastal aquifers carries a diversity of bacteria able to grow at least on marine zoovel agar plates. So it is possible that at least a fraction of the marine seed bank is hidden within coastal groundwaters. So there are many remaining questions, many open questions, such as what are the limits of microbial metacommunities, if any, what is the fate or relevance of dispersed microbes, what are the ecological implications of rarity, and whether we can delineate the actual size of the seed bank. And indeed, many of these questions may remain unanswered because we are slowly moving from amplicon sequencing towards techniques like metagenomics that, at least for now, have a more limited capacity to detect the rare taxa. So almost 20 years later, it seems that this pressing question of Carlos is still quite difficult to address. So with this, I finish this other open question. and I think I could finish just thanking all the people involved in this project, Carlos for bringing all of us here, the organizers, and you for your attention.
Oh, thank you very much for this presentation, Pep and Clara. And the next speaker today is Silvia Cines. She's a scientist here at the Marine Science Institute, and she has worked with Carlos from many, many years ago. So, and she will talk about the beginnings of bacterial diversity of the studies.
It's very nice to be here. To me, Carlos is one, I will say, one of my mentors in this long career. But I want to start from the 90s. Not only is this the best music and the best cinema, but also this world is when everything started in microbial ecology using molecular approach. So I think most of us in this room, we know already that most of the biomass in the planet and in the ocean is microscopic. It's invisible to our eyes, and is constituted by the microorganisms of the plankton. Indeed, about 90%, between 70 and 90% of the total biomass in the ocean is constituted by the plankton. And when I say plankton, I'm referring to microorganisms, little microorganisms that goes to the protists, to bacteria, bacteria, fungi, and viruses. And let me see why it's not working. Hello. Well. All right, there we go. So my career mostly has been focused on bacterioplankton, and it's one of the components of the plankton. And why? Because first, there's many, many cells in the ocean. There's about 1 million per milliliter of prokaryotes, I say bacteria and archaea. And when you extrapolate that to the total volume in the ocean, it means that we have a total of 10 to the 29 cells in the ocean, which is a huge number. And also, we know that at least half of the primary productivity in the ocean, a significant fraction, is thanks to the photosynthetic bacteria, which I'm calling cyanobacteria, Procorococcus, and Ectococcus. And also, most of the respiration in the ocean is also microbial, and bacteria and archaea are key components in all biochemical cycles, the carbon, nitrogen, sulfur, everything. So, but how many microbial species sit in the ocean? So the last estimation is from this paper is 10 to the 10 species, which is insane. So that's now we want to go back in time and see how we can really start at that time in the 90s, how we're starting to realize that there was a huge diversity and we need to identify, and I didn't know how. So for that, I highly recommend that you read this book, the last, Carlos' book. Yesterday was the presentation, it was really nice. And he has one chapter that is so beautiful written, like the whole book. This is a microorganism, I highly recommend it. So first, going back to the 90s, you remember the people who that was in this world, because I see many, many young people. And that in the beginning everything was on isolates, but just remember in the 1986 was the beginning of what we call the molecular phylogeny. What Carl Wood and Norman Page have this the tree of life based on the 16S ribosomal genes, and also it was a very great moment with isolation of the most abundant photosynthetic bacteria, the Procorococcus by Penny Chisholm. Also in the 90s, in the beginning of the 90s was the first discovery of the SAT-11, and also the discovery in 1992 of marine archaea by Delon and Jack Furman. And also a few years later, also we moved from knowing that this bacteria was a really a key bacteria in the ocean to realize that it was actually the most abundant bacteria in the ocean. And also it was possible in 2002 to isolate this really high abundant bacteria. But in this moment in life, when we were analyzing microbial diversity, we basically could analyze about 100 sequences. And this was a milestone. So this is, that was the beginning. So in the beginning, it was not metagenomics. In the beginning, it was not high-throughput sequencing. In the beginning, the way that we do it is just extract the DNA and PCR amplify usually the gene marker that for diversity was, for prokaryote was 16S, for protists was 18S. And then it was like a two approach. One was what we call fingerprinting approaches, and there was the GGG and restriction fragment length polyforming. This is the one that I used during my PhD. And it was also a cloning and sequencing approach. So in the beginning of my PhD, I want to keep in mind two things. One is the number of the authors here. This is my first PhD paper. As you see, the last author was Carlos. It was not my PhD director. So actually, Paco was my PhD director, but Carlos was the last one. And so also you realize there's only 3 authors. This is a change dramatically with all the papers we have now, because now the science is multidisciplinary. Before it was just focused in one particular sampling on transit or whatever. And this is me with my dog and a lot of waste biomass. So this is the first paper. Basically, what we did is Paco told me, okay, you have to go to Barcelona because these guys, they do always campaign. They have many very nice sampling. And so I went to Barcelona, and there was this campaign, France, 1995. And with Carlos, we also started to think, we were interested to separate the free-living bacteria and also the particle-attached bacteria in that paper. So we was planning to do a specific safe fractionation protocol that's different with the ones that we're using now. And And so then I went to Barcelona. That was my first time that I met physically with Carlos, that, you know, it was in the other building, and also met Pep, Ramon, Dolores, and everything. And so I realized how, you know, well, you know, organized these people were, so how high level the science was, and I was really impressed at that time. So this was one of the restriction fragment length polymorphism. So basically, you amplify the cystinase of the whole community. You digest. Usually, you combine different enzymes, and you run this gel with polyacrylamide. And I can tell you that this gel was like this. So, and then you compare samples, the absent of the present absent. So, and you do this dendrograms on similarity. It was not phylogenetic at that time analysis. But it was interesting because already we can see that using these restriction length polymorphism fragments, we could separate already, you know, the bacteria from the particle attached, that was this cluster, versus the free living that was here. And also it was interesting that we saw the samples from the coastal were more related than from the slope and from the deep, that which means this is coastal slope and the deep here. And also the vertical from surface to deep. So you see that the deep sample was really far away. So you can see really some patterns already. And this paper to me was one very, very important because it allowed me to select, you know, few samples, very key samples from these different clusters to go to the next level that was actually do environmental clone libraries. So I passed the next 2 years to analyze 120 clones. So there was a lot of work, I can tell you. So, and with this analysis, there was, you amplify the genes and you clone the cystinase in plasmid and you sequencing. You could see like a different phylogenetic clusters because, and we realized already that Alteromonas is one of the key members on this community of bacteria attached to particles. So that was in 1995. Probably this is one of my best paper of the best paper of the PhD. And many of you will remember that published and applied environmental microbiology was the best. At least to me was the, the best at the moment. So time passed. So there was the next decade from the 2000 to 2010. They arose the microbial the diversity by using high-throughput sequencing and the omics. When we call high-throughput sequencing, when there's different strategies. For instance, one is amplify one particular gene that usually is the 16S or 18S. This is what we call metabarcoding or amplicon tags, but you can also amplify functional genes like NFH. But also, there's another strategy, there's do metagenomics. That's the way we do it very often now. But was mostly based on the DNA structure in the community and then sequencing the DNA fragments from this community, but without cloning this fragment. And then you can extract, because you have all the pool of the DNA fragments from the microbial community, you can extract as well the 16S and the 18S from this environmental metagenomics fragments. So this paper to me was crucial, and I was in 2004. I was already my postdoc in Boston. So I saw this paper with Craig Bainters, and many of you will remember. And it was interesting because they didn't do exactly metagenomics. They did what is called whole genome sequencing. Basically, they have this fragment of the DNA from the whole community, and they clone. They clone it. So they cloned 1.6 million DNA fragment. That was insane. And they discovered 1.2 million of unknown reads, which means that most of the diversity was unknown. And from these millions of genes, they extract only 1,000 whatever different 16S fragment. And from those, they extrapolate the number of species. That was 1,800 on the particular, Sargasso Sea sample. And at that time, in the same year, we were published with using a different strategy, a similar number. So basically, I spent the next 3 years doing the analysis of thousands environmental clone libraries. So still we were doing clone libraries. But using this huge, at that time, because this takes a lot of time, we also come up with a very similar species number. That was 1,600. And also in our paper, we observed that half at least of the diversity was related between closely related taxa, and it was called microdiversity. And this microdiversity was not due to bias or the tag polymerase or either the different copies of the cystines. So that was in the same year. And also 2 years later, there was as explained now beautiful by Clara, this paper with the contents of the raised biosphere. And they used in high throughput sequencing, and they sequence like a 2 orders of magnitude more. So more than 1,000 environmental sequencing, they analyze 100,000 PCR amplicons of the cystinates of these communities. And they identify about 5,000 taxa, and they extrapolate of this number of species, 18,000. Again, okay. Come on, Jesus Christ. No sé, it's blocked. Uh-oh. Sorry. A ver. No sé cómo funciona. No gira.
Ay, Microsoft.
Microsoft.
¿Cómo salgo de aquí?
Con lo bien que iba, cachi la mano un tiempo.
S'ha quedat penjat.
A ver, igual es el cable o qué.
Espera, no, pues es este.
Okay. So we coming in. Woo! So Carles, in the same moment that this paper wrote this beautiful, opinion paper on this marine microdiversity cannot be determined, and Clara has already explained it. But he, I think, oh man, okay. Wait, sorry. Jesus Christ. No, para atrás. Okay. Okay. So he explained beautiful the concept, and he makes this concept of the rare biosphere, and where the most abundant taxa, this is a rock abundant curve, that was more prone to be actively grown and under pressure by predation and hydrolysis, but it was at the same time the main contribution to the different biochemical cycles. And then this rare biosphere, that it was a key role for as a seed bank for increasing microbial diversity. He also mentioned that to target these two different, the abundant and the rare, different techniques are and exist. The molecular techniques are the ones that we probably will reach more the most abundant taxa, but the culture mostly were those that target the rare biosphere, even though that we know that some culture represent abundant taxa. So now that he didn't listen, as I think Carlos became a little obsessive about the rare biosphere. From the following years, it was rare biosphere, until even 10 years later, from the first 2006, he said, okay, I want to test this rare biosphere. I wanted to prove that I can target, so how can I do that? So I will spend half of my Plan Nacional money and make this experiment to really sequence as much as we can, with 2 samples from the surface and DCM, and amplify like 500,000 of tags, amplicon tags from these 2 samples. So they realized this is a paper by Viviana and Carles, led by Carles. So these 2 samples, they observed that the number of OTUs, the taxa, in the surface was way less, and it was reaching the plateau, but in the deep ocean it was higher, and they estimated that it was around 1,600 species in the surface and about 5,000 in the deep. So again, this number was similar to other estimations using different approaches. They also compared the amplicon tags, so the environmental samples, with the isolates that they collected and this environmental sample from the surface, and they realized that the cultures usually belong to the red biosphere. They couldn't get any culture bacteria that was highly abundant in the, and matched with environmental samples. But I think we all can agree, and I think Clara did a very nice explanation, that the red biosphere knows only those bacteria that can always be in red, and this is a strategy to be red with a slow grower or dormancy, but also there's also taxa that can become abundant again if the right conditions exist. And this is something that we started also to explore using the deep ocean data set from Malaspina. There was this is a paper from Guillem, and that was co-directed by Pepe and myself. And in this data set, they're using this ampli contacts from 32 stations from the Batipelagi for 4,000 meter depth. And it was interesting that we do this rank among curves. The 10 most abundant taxa from the Batipelagic deep ocean was, you see here, some taxa related to isolates, Alteromonas, Lomonas, Erythrobacter, Pseudoalteromonas. There was a kind of shock for us because if we do the same curve from the photic layer, it was all uncultured, the taxa. So we wanted to test that. So the way that we did is to compare all the environmental data set from Amplicon, from Tara and Malaspina, from surface and deep, with a huge collection of isolates, also from different depths. And this is a paper that it was published by Isabel, also a PhD student from the group. And it was interesting because you see this is the isolates versus the Amplicon tag. This is a Amplicon sequence variant. And if we compare the isolates against the Amplicon tags from the photic layer, this is the colored dots means that a perfect match, 100% identity between the isolates and the environmental samples. And you see that most of the isolates were here as a red biosphere. But when you compare these isolates with the environmental data set from the deep ocean, you see that some of the culture were really become abundant, very abundant, and it was actually between the top 10 most abundant taxas in the Batipelagic ocean. So even though the number of richness was not really high, the percentage of the reach that this isolate recruit in the Batipelagic was really high. It was up to 30% in the free living, and the particle attached was even higher, 45%. And also, it was beautiful, this paper that was recently done by Marta, Sebastián, and Pek Gasol, where they prove that also a small percentage of the surface Amplicon tags that exist in the Batipelagic, those that exist have a huge contribution as an active member on the Batipelagic microbiome. So it means that a very rare surface taxa contributes highly as an active member in the Batipelagic microbiome. So we pass now from the transit local sampling to the global ocean exploration, and it was, I think, in this moment that it was really nice because it was like a different initiatives. One of these international thesis of marine microbes before was the global ocean sampling, and of course, Tara Ocean and Malaspina. Actually, Carlos was involved in the international thesis of marine microbes, and that was the first estimations of combining a lot of samples from different habitats, marine habitats, from pelagic and bentonic, oxygen minimum zone, and they amplify like 4 million of Amplicon tags, and they reach to this level of species, about 65,000 species. Then with Tara, Malaspina, rights, we use a lot metagenomics, and this is just to remember a little bit, there was very complementary because Tara was between 2009 and 2013, and Malaspina was 2010. This was in 2 phases, this is the beginning, and this is Tara polar cycle. And Malaspina was in 2010, so they covered complementary regions and other totally different regions that was not covering by Tara, and also reaching down to 4,000 meter depth. That Tara only reached up till mesopelagic. So we use we extract from the metagenomics the cystiners fragment, we call it the M tags or my tags because it was Illumina sequencing at that time. And there was also an estimation about 3,000 species globally using all this data set. In Malaspina, from the deep ocean, we have a small data set for 4,000 meters depth, and the number of species was less, also using amplicon tags with a bathypelagic data set from Malaspina. So we have already some numbers, and also the nice part of this global expedition, it gives you an idea of the diversity change across vertical profiles, but not using one single transit, but more globally. And this is on Tara, how you see already increased with depth from surface, DCM, and meso. This is already proved by many other studies with local transit. And in Malaspina, we recently include also the bathypelagic that was not included in Tara. Also, we see this increase of diversity with depth. This is the last paper of Pablo and Felipe, using Malaspina vertical profiles with a nice data set. So, but again, metabiology allows us to go beyond just to explore the diversity and functional, because we can understand a little bit the metabolisms. And I think the nice part of the metagenomics is that we can now reconstruct genomes from the whole pool of DNA fragments. And just to comment a couple of the more examples that also collaborate with Carlos with the using, I think to me, it's one of the good approaches to go to explore microbial diversity at high resolution at the genome level. And we were able to reconstruct MAX from the Tara polar ocean cycle. And again, this is a work from Marta Royo that is here. And I think I want to mention that it was co-directed by Carlos and by me, but actually it was a fellowship that it was funded by Carlos. So again, his generosity allowed me to co-direct Marta with him. So he was involved, Marta, in doing this nice work with the Tara polar cycle, with the reconstruction of these 500 mass with the help of Pablo. And from those, we realized that most of the diversity was unknown at the species level, and more than half of these genomes have a polar distribution. We also explore the prevalence of the mesotrophy in the polar regions, and also we were able to identify where we say that it's a polar sentinels, which means our genomes that are highly active and have a polar distribution. So that was a nice moment, also collaboration with Carles, and also the collaboration is not past, but is future and ongoing, because this is our new project that was funded this year. We didn't start, this is polaromics, this is biogeography, and we explore the adaptation mechanism for prokaryotes from different biomass. And you can see this is the team, and Carlos is involved. So I hope that Carlos can join us in the campaign and maybe write another book for Neves. And just to finish, in this project, we're planning to use all the approaches, not only the isolates, but also the metagenomic assembly genomes and the singly amplified genomes to really have an holistic view of microbial diversity. And just I want to finish with this slide, was going again back to these questions. So how many species are in the planet? 10 to the 12. How many species maybe in the ocean? 10 to the 10. But we are far away. But this is the last a nice paper of in nuclear research that just came in, this month, led by Pierre Borg. And with the inclusion of more than 1 million of MAX from different biomass, from different 70 biomass different, with more the integration of more than 700 studies, and more than 100,000 different microbial metagenomes, and they explored the claim with this number with almost 100,000 microbial species. We're still far away from those numbers on the top, but we are every time we are increasing the number of microbial species. And just to finish, just to thank Carlos for all the joy that within science, but also outside science with my family, with Juliana and Nerea and Cristina as well. Thank you.
Okay. Thank you very much, Silvia, for this presentation. We should move on. Now the next speaker is Daniel Bolot. I just use this talk to remember that protists are microbes too. So in this talk, no? Protists are microbes too, basically. Too. So, and again, Carlos has been involved in the initial explorations of the pico-eukaryotes. So in the exploration of prokaryotes, he was participated, but he was seminal in these studies, and that they were done together with Daniel. So this is why he's here today. So that comes back to there was a project that Carlos and Daniel. I don't have to.
No, because I'm going to.
I cannot introduce anybody because then.
Oh, yeah, I just go.
You just go there, and there is... ¿Te has quedado, Silvia, el mando?
Here.
Here.
Okay.
But you first start like doing that. Sorry, the pointer. Yeah, and then it appears. So see, it's the same size of the...
Okay.
It's a pico pointer.
Okay. Well, Pep and Ramon, thank you for the invitation. As Ramon said, when he sent the invitation, I say, okay, I used to be in Singapore for 5 years, but now I'm in Europe, so I say, okay, I'm coming, because I always like to come to Barcelona. So I'm going to talk about protists more and phytoplankton even more. And yeah, I start with this picture of Ramon and Carlos looking for phytoplankton in the Arctic. So when I prepare this talk, what I say, okay, I'm going to talk about the contribution of Carlos in phytoplankton, but eukaryotic phytoplankton, and then I do things that are, it's not bad, it's not good, it's like stalking people looking on Google Scholar, okay? So I look at the most cited papers from Carlos, and I was thinking of Carlos more like a bacterial person actually, but then looking at his contribution, his key contribution is, some of his key contributions, there is this one that was mentioned earlier, but some of this key contribution is, and I go back, yeah, are about pico-eukaryotes. So I'm going to talk about this, setting some background first, and then talking about these PicoD projects that we did together, which is a very, very good memory to me. So first, I will give you the background, which is about the same that Sylvia said mentioned for prokaryotes, for eukaryotes. People had a very simple view of marine food webs before the 80s. It was very linear. You had phytoplankton, big phytoplankton eaten by copepod, that was eaten by fish, and fish were eaten by bigger fish, and up to this orca, or us actually. I think we are worse predators than this orca. And phytoplankton was thought to be dominated by large cells like diatoms and dinoflagellates, because that's what you could see with a microscope. However, there were some areas in the ocean where we knew from a long time that there was very low biomass, and that this biomass was concentrated at depths. It was not in surface, it was at depths. So there were a lot of theories about calling it detrital chlorophyll, or so there were a lot of theories made, especially I remember before I began my PhD, which didn't make sense to me. And then there was this discovery by John Waterbury in Woodsall of Synechococcus, which was a small unicellular cyanobacterium that it could detect with these techniques that at this time just arrived in microbial ecology, which was epifluorescence. This was a technique that had been used before, but in microbial ecology, it really appears at the end of the 70s. And more importantly, was able also, because he had done his postdoc at the Pasteur Institute, to culture them, so which I think is one very important factor to study this microorganism, is to put them in culture. But there were not only cyanobacteria, there were also a lot of eukaryotes. And this is a unique study, because there have been very few studies like this doing electron microscopy of marine samples. And what they found is that they found a lot of cells that were, there were some prokaryotes like Synechococcus that had been discovered just before, but there were a lot of eukaryotes, and you can see because of the nucleus, because of the chloroplast, and because of some structures that are not prokaryotic. Also, at the same time, it was found it was not only the biomass, which was a very small fraction, but also the production. And this is a work of Billy and Trevor Platt, where they found that in the tropical ocean, more than from 25 to 90% of the biomass was in the small size fraction, and that 20 to 80% of the carbon fixation was also in this very small fraction, less than 1 micron. One fraction that we don't use so much now. We use more 3 micron, but whatever. So, and then this led also to the discovery of what we know to be the most abundant cyanobacterium now, which is a Prochlorococcus. And this was discovered through another technique that became prevalent, and of which Pep is one of the master, flow cytometry. So this led all this to this concept with the bacteria also that Sylvia mentioned, to the microbial loop, where instead of having a linear system, we have something which is more of a circular and very complex system. And this was really changing completely the paradigm in a few years, actually. But we could still not, I mean, it was very difficult, even for bacteria, to know what was the nature of the bacteria. And that's this paper for me, when I read it, I remember very well, I said, wow, wow, this is a way to go, actually. For me, it was really, I remember very well, I was in Hawaii, and then this was published, and then I looked at it, and I said, oh yeah, we need to do this. Leading to the discovery, of course, of all this lineage of bacteria. But for eukaryotic phytoplankton, the situation was a bit more dire than for bacteria, because with flow cytometry, we could see Prochlorococcus here, the Synechococcus, and then we had a cloud of picoeukaryotes, but we had very little way of telling what they were. There were very few species described at that time. In the early, just at the turn of the century, Micromonas, which had been discovered in 1952, Batycoccus, small green algae, both of them, Pelagomonas, which was a Stramenophile, Ostracoccus, another green algae here. But very, very few, and it was very difficult by microscopy or flow cytometry to know exactly what they were. So that's when we did together this PICODIF project, and it was a European project which were still of human size, because now European projects are not anymore of human size. We had, and I put this slide because at this time, I know you probably don't remember all, but we love blue slides. It was always to have a blue background. So this is original blue slide, and where we had 5 partners. So I was coordinating this project from Roscoff. There was Dave Scanlan from the University of Warwick, a specialist of cyanobacteria. Linda Medlin from the Wegener Institute, who was applying already molecular methods. Then Carlos and Ramon from Spain, from here. And from Norway, we had Jan Trondsen and Wenche Økrum from the University of Oslo. So this was a small group, but I think we got very, very active, actually. And our strategy was to get phytoplankton data on diversity from culture and clone library. And we always put culture and molecular method together because I think they were very complementary. There is the issue of that we don't isolate everything, and there are a lot of things that we still cannot isolate. And then we were modest in our field work. We were just looking at coastal diversity and trying to look at annual change in diversity. And we had a few websites. We have a website, field sites, sorry, not yet. Well, we had a website, actually. I didn't find it. It's lost completely. That's the problem with the internet. It's lost. Before I could find it in these archives for internet, but I think it disappeared. So we had Helgoland, this island that's sampled by the Alfred Wegener Institute, Roscoff, of course, and Blagnez. And these sites actually now have been continuing. And at least for Roscoff, and I think for Blagnez, it was the same. It was the start of this annual series, actually. So there have been continuation of this project. And then we had some crews, but we didn't use so much of data from these crews. So I think actually some of this work predate Picodiv, but we sold it as not yet done in Picodiv, like always do. Use a project that you do before on hidden funds and then put it in the next project so that you have already deliverable like 5 months after the beginning of the project. And for me, this is the key of what happened after with eukaryotes. It was cloning, so do what Giovannoni did, but it took for eukaryotes 10 more years than doing it for and I don't know why. I think nobody had the idea, actually. So this first paper was, so Carlos is a co-author, and this is the same team, actually, that you mentioned where you did your PhD. And Puri now is in France with David Morera. And so when they did this clone library, what they found, actually, this is from the Antarctic. They found a lot of new groups, a lot of sequence which are nothing to do with available sequence, and especially they found these 2 groups of marine alveolates that we know now are syndinyales, or they're parasites, but they're probably one of the most diverse and abundant group in for eukaryotes in the ocean. You find always them, and there are so many papers now saying, marine alveolates. So what was really amazing is that at the same time in Roscoff, we had exactly the same idea without ever talking to each other before. So I had a postdoc from Korea, and she did exactly the same strategy, so sequencing like 100 clones, because that's all what we could do. It took a year to do this. And, but what was very interesting, it was this time from the equatorial Pacific, so completely different ecosystem, and we found exactly the same thing, many new organisms, especially in the alveolate group, but we were much more cautious, but not as brave as Carlos' paper. We said question mark. We did not know what they were. She was, Sangyo, she was very concerned about chimeras, whether it was chimeras, because when you have a group like this, so we did not know what it was, but this is marine alveolates. Oops. And also we found a lot of new sequence from green algae, which are then only present in equatorial water. And now this is a new class of algae called Chloropicophyceae. What was amazing is that these two papers came out. So our was submitted before to PNIs. And this was reviewed by Norman Pace, one of the big microbial ecologists in the world. And he said, this is not new, you have nothing new. This is known for bacteria for a long time. So he just slashed up his paper, our paper. And I think I remember with Carlos, we were in Chile when Norman was there, no? So yeah. So I pushed him a bit because I think I poked him a bit because this was. So anyway, these two papers were back to back in Nature issue. And at the same time, there is another paper that Beatrice also authored, and again Carlos, of course, and Raman. And Carlos always telling me, yeah, this paper is as good as the two other papers. It should have been in Nature as well, but it came a bit later. So they use the same strategy from Mediterranean Station, Antarctica, and North Atlantic. And what they use actually is using DGGE to separate the different clones, and then being able to sample more accurately, actually, the clone libraries than what we did. And what they found there is this novel straminopause, the famous mast that now is the shop of Ramon. Anyway, so I think these three papers, they appear together, and they were really founding the base of this eukaryotic diversity. We also, during this project, developed fluorescence in situ hybridization, which allowed, by using probes, to look under the microscope and count, actually, the different organisms, because like metabarcoding now, clone libraries are not really quantitative. And this was done, this is a cruise done in the North Atlantic, I mean, between Norway and Svalbard, where we were able to estimate the abundance of Micromonas. I think that Connie will talk about Micromonas. That we know now is a Micromonas polaris, probably. Again, another paper from that time where Carlos is a co-author is then looking at culture, because as I mentioned to you, it's very important also to isolate. So in that paper, they isolated a lot of cultures, new cultures from different groups, and again, this is a key contribution. During this project, I think there was a lot of things that then became quite important for photosynthetic eukaryotes, like I mentioned the mast. So this is a tree by Fabien Burki, one of the latest tree, and in blue you can see the groups that were investigated during this project with Carlos. So we have some groups which are either straminopods or alveolates, but also we have some groups like Picozoa, which we thought initially were photosynthetic, but now they seem to be purely heterotrophic, which have quite interesting place in the tree of life. So two of these groups is Picozoa and Telonemia, and they have kind of unique place in the tree of life. We also discover new classes like Chloropicophyceae, and also new species and orders. So this was both molecular but also looking at culture. So, so now, where are we? We are quite far. As Sylvia mentioned, there is these big expeditions. So we were only sampling a few places, but now there is this big expedition like Tara or Malaspina. And we have gone with high throughput sequencing to using what's called metabarcoding, where we can get so many more data, and we can do some papers where we do some analysis where we look at diversity and so forth. But for me, one of the most interesting advance of this is doing biogeography and at a larger group, but also at the species level. And this becomes very, very interesting, especially, for example, for polar species, to look at the change that are occurring, and probably Connie will mention this. But also for eukaryotes, we can do metagenomes, and I like this title from, so this was a paper from Tom Delmon, where he did a reconstructed metagenome assembled genomes for eukaryotes, where you can finally look also at functional clustering. And in contrast to prokaryotes, it takes much more resource actually to do this. 1 minute? Okay, I'm good. But this is probably a way to go. We have new paradigm. I think this is a very big change now that we look at many more processes like symbiosis, mixotrophy, parasitism, predation, and we have many more tools to look at this. So there is a flurry of studies now, especially parasitism and mixotrophy, which were not considered before. I'll finish with this, with some pictures from this happy time of Picodive, where we were we had a lot of meetings, and it was always very, very nice and very friendly, because it was a small group, so we knew each other all. Thank you again, Carlos, for all your contribution for the eukaryotes. I think this was...
Many thanks, Daniel, for this very interesting talk, for this visit to the eukaryotes. Now we keep moving in this session, and the last talk before the coffee break is going to be by Jeroen Pinassi. He's a scientist at the Kalmar University in Sweden, and I think we are going to move now through a little bit more complex molecular and sequencing environment, which are the genomes. Many of the things that we have seen, or part of the things that we have seen, have been studies on diversity using a single gene, but now you can get to the genomes from these species once they are cultured, so we can go to the genomic surroundings.
No trouble. Aquí. This thing, and the pointer, you start right here, and at some point, if you do that, at some point, yeah, it appears.
Good. Thanks a lot for the invitation and for the possibility to be here. I'll start off with a nice little bird for you, Carlos. This one is on Öland in Sweden right now. It's a desert warbler from Kazakhstan that turns up sometimes in Sweden. Little brown jobs, I know, is one of your favorite things. and this one can, well, it's a desert warbler, but it's a nice dessert too. I came to Barcelona to work with Carlos and the team, and Celia and Pep for sure helped out a lot in the attraction to come to Barcelona to learn molecular biology techniques that were modern at that time, DGG and cloning of 16S sequences. But I had worked with a lot of cultivation things, so I couldn't really stop putting seawater onto agar plates. Just from these experiments that we were doing together, I plated a bit of water. And for example, this orange and yellow, these orange and yellow things will appear a little later. They have very nice pigments and also happen to have some other interesting genes. One of the first things that we did, or that Carlos invited me to join an experiment in Alabama, in Dauphin Island, a mesocosm experiment with Rafael too, here you can see. And this actually, it was important because we made a nice experiment, but it was also very important because it put me and us maybe in contact, close contact with Jose Gonzalez, which we have never met, but we kept the line open on the phone, actually. And Carlos often commented, it's so good that you call on the phone and not only try to email. And so email is complicated, but phone is nice to use. And Jose will have a very important role here in exploring the genomes, because what happened was that a few years after doing that agar plate, it turned out that an offer to sequence the genomes of marine bacteria, and I think this is a huge turning point for marine microbial ecology in general. So in 2003 there was a call, and then we proposed to sequence some 15 bacteria from the Blanes time series and from the experiment we had done. And 10 of them actually were selected for sequencing, which turned out kind of interesting. And then Carlos says, let's do some, to work with this, let's do an annotation jamboree, because it's the only way I see to get started on these issues. I mean, to really, I mean, how do you start with working with genomes? We know they have thousands of genes, each bacterium, and now we got 10 genomes at the same time, 30,000 genes to look at. What do we do? Well, Carlos suggested, let's sit down, let's look at the genes one by one. Let's make it a group effort. We each have some 300 genes to look at, and we report them to Jose, because he kind of likes putting things together. So I talked with Jose the other day, and he said, yes, Carlos was very diligently doing this every week, reporting these 30 genes as it should, and Jaron, you did not. I had to remind him, and well, I can maybe live with that. But anyway, this was the way to go, little piece by piece looking through the genes. And it turned out to be the one we focused on was a Polaribacter. I'm still trying to figure out why we did that one, if it was because it was beautiful, or we had some indications it was a bit abundant sometimes in the ocean. Getting this kind of jamboree thing going, I think it gave us a bit of taste for it. It was a nice thing. Carlos was very driven on this. Never ending, discussing what are the possibilities and how can we continue with this. And which resulted in a very nice paper where we compared the genomes of 3 different bacteria, because 3 different bacteria is kind of 9,000 genes to compare, which is nothing compared to what Sylvia showed with the 530 genomes. But let's bear with me a little bit. And of course, we think this takes time. It's many genes to look through. And when Carlos had done the revision of the manuscript, he was very telling in a mail to Jose, don't worry, now at least Jose can sleep well. So, because it took time. Jose can be a bit impatient sometimes. Impatience is not really what we know Carlos for, but sometimes his patience runs out. So, for example, when he had to refill all the author details in the food web, in the submission page, yeah, I don't need to read it. And another piece of story that the same genome sequencing led to was that some of these bacteria that were genome sequenced turned out to have the rhodopsin gene. And it was scanning through the list of genes just to see, is there anything in these genomes, because we didn't know anything about genomes. Is there anything that we think is interesting in these? And then it turned out that two of the isolates that we had annotations, automatic annotations, that we kind of could recognize a little bit. And then Jose commented about the identity of these genes in 2005. I get very nervous from seeing this. Okay. Yeah, because he kind of had a hunch what this is leading to. And then Carlos, enthusiastic, I like this with genomes more and more every day. Oh, fantastic. It was just a couple of hours later from Jose's email. And then the next day, when Carlos had time to think a little bit about this, he said, wouldn't this make it for a nature story? And then I should take a step back because not everyone here has kind of grown with the rhodopsin stories. So rhodopsin is a membrane protein. It uses light to pump protons, and those protons, the electrochemical gradient, can then be used to synthesize ATP. And there is a little funny thing here, is that the key molecule here is retinol. It's a carotenoid. And this is the reason why because we have these proteins in our eyes. So this carotenoid, the reason why you should eat carrots is because it helps you replenish this little molecule. But that's a side story. So all of us know rhodopsins in a way. But the question was then, now that we had this gene in a bacterium, it had already been described that this was present in some 60 or 80% of marine bacteria in the surface ocean, but no one had shown that it's actually functionally used to get energy. So as Carlos suggested, let's make the experiment, see does it work? And then, so we did seawater culture with one of the rhodopsin bacteria, and in the dark it didn't grow. In the light it grew very well. But then we had the problem, what do we do here? How do we know that the dark ones here are not actually dead? So we had to redo the experiment a couple of times, but with cultures where we had the cultures in the dark with aluminum foil around them, and then took it away, the aluminum, so that light could come in, and then we could see it. Okay, it wasn't that they were just dead by some mistake or something. It's actually that these bacteria can use light to grow. And that became the requested for nature story. And then we were discussing when in the first submission, what should we put in this? And I was strictly thinking about these biochemistry figures and stuff. And then Carlos said, yeah, but they like colors. Let's put some photos of the bacteria because they are beautiful. And I don't know how much it influenced, but for sure it gave, we made it. And I think it was, so this science is not always this strict science. It taught me that color and beauty is part of what makes a good story, maybe. And then to show a little bit more of Carlos' then inputs, we had continued to do some gene expression analysis. This is the rhodopsin gene expressed in the light, but the black bars, you don't see them. It wasn't expressed in the dark, so it's really nice. It's used in the light. And then we were very enthusiastic that isocitrate lyase is a gene in the central metabolism. It's really in the TCA cycle, so it's something that is quite unusual, quite usual. And we having the connection between rhodopsin and really central metabolism, we thought this was marvelous, and had written the manuscript and then turned it to Carlos to get some input, and then we kind of thought Zaragoza here means something, it's essentially incomprehensible, maybe, if I'm a bit rough on the translation here. But the discussion wasn't much to understand. But with some good help from Carlos' patience on working with words, I think it made a nice story in the end, where actually rhodopsins, no, it's not just some auxiliary little subset of gene that is kind of for something, but it's actually linked with central metabolism of these bacteria. And then, I mean, there are so many stories, and I think several of them have been told already in the morning here. Carlos has continued. I noticed you continued with Polaribacter. It's an interesting one, because I guess because you've worked with it before, but also because it is actually quite abundant, especially in polar areas. And it turns out that this Bacteroidetes here, this Polaribacter, it actually takes up iron using transporters, but it's a cheater. It doesn't synthesize all the siderophores, the chemical capturing mechanisms to do it. It's cheating. It's taking up iron that other organisms have kind of struggled to condense into a format that can be taken up. And Sylvia mentioned this, but it's interesting how cultivation has come back in fashion. And I think by the time I came to Barcelona, cultivation was so out of fashion. It was so not the right thing to do. And I was quite convinced that it would never, ever come in fashion too. But I think the circle is kind of closed, because thanks to the molecular biology and the metagenomics, there has been a huge increase, I think, in the interest in isolates and having model organisms to work with. So I think what I guessed would be that no one ever would turn back to isolates. It's actually proven wrong. I think model organisms now are much more appreciated than some 20 years ago. And Sylvia also mentioned this, and if one genome wasn't enough, here Carlos is diving into over 500 of them. Maybe you didn't report 30 genes a week to get this going. And more genomes coming, and I don't know how many weeks it took to sort these things out, and I'm not going to go into the detail, other than that it allows you to use genomes now to see what are ecological units, what are ecologically relevant gene systems that are maintained by some taxa, discarded by other taxa, and how does that relate to the ecology of bacteria. And with that, I'm looking forward to your coming endeavors, Carlos, and several of you in the audience, I'm sure, will pitch in on that too. Thanks. I can just say this is a white-billed loon from the polar Arctic. It's one of the target species when one goes to the Arctic.
Thank you very much, Jaron, for this fantastic talk. I think now we have time for a coffee break, and we will reconvene here at 12. So 10 minutes later than we said, but 12 sharp.
So I go upstairs to the patio, and it should be there. If it's not there, we can follow them.
I hope you had a relaxing coffee, but now it's time to start. Thank you. Okay. So the next, I think we should move because we have still a few talks to do, and also Carlos asked us to talk a lot, so we have to give some time for him. So the next speaker is Connie Lovejoy. She's a professor at the Laval University in Quebec, but she was also here as a postdoc 20 years ago. And she will talk about polar research.
First, thank you very much for everybody here. This is a great turnout. I was asked to talk about the polar regions, and I decided that I don't know that much about Antarctica, so we're going to stick with the Arctic. And in line with Carlos's other literary career, this is the Arctic chapters. Okay, let's go this one. Okay, I made an outline for this talk because that's the only way I can focus. And basically, my whole career has been following currents and leads. And currents are what's in the ocean, and leads are people like Carlos. And also, in the Arctic especially, you have these leads that open up in the ice. And as Leonard Cohen, my other favorite author, said, you need cracks because this is how the light gets in. Oh, and then I'm going to talk about Micromonas, and basically I have two stories, and the second story is going to be a short one on Acantharia. Okay, first of all, this is a quick review. Thermohalide circulation, the water gets formed, it's deep in the Arctic. When ice freezes, it ejects brine, and that winds up on the bottom of the ocean. And this is why, probably one of the reasons why the deep sea bacteria are kind of related to each other. And also there's a lot of time to make diversity in this whole system. And there's both in the Arctic, there's two places where you make deep water formation. In the Antarctic, you form deep water. But that's not really what I'm going to talk about. We're going to then move to the actual Arctic Ocean, where you have water that comes in on the Pacific side over here and kind of zooms along here, where we did a overwintering expedition with Carlos in here, the people from Barcelona. And then there's this Beaufort Gyre, where I've done had a lot of research cruises. And then the water from the Pacific actually will come back down here and go through Davis Strait, Baffin Bay, things like that. But basically, all of this water kind of works its way through the Canadian archipelago. And the nice thing about this one section here is there's only one source of water, surface water, that comes in, and it's been influenced by the Pacific, and it's been in the Central Ocean for a while. Then, of course, you have some water here that kind of goes up that side too, but we won't get into that right now. So that's your quick review of the current system. And we're going to go back and talk about my beginnings. I had a different life. I was born the same year as Carlos, but I got back into science seriously got back into science in 1997, and did my PhD actually in the North Water, which is between Baffin Island and Ellesmere Island. And at one point, I had frostbite, and they flew me off the ship, and I quickly put a bunch of things I'd had culturing in a refrigerator in the back of the ship. And because I figured, at least if I can isolate some algae, I might have a chance of saving my rebirth in science. And in amongst those was this Micromonas, which Daniel mentioned, and it was isolated from the water column. And then after I finished my PhD in 2002, this is when I started my postdoc here in Barcelona with Carlos and Ramon Massana. And came here because in 2002, after I finished my doctorate, I went and did a cruise in the Canada Basin. In fact, it was two cruises. I was one on Louis Saint Laurent, and the other one was on the Marais, and switched ships in the middle of the ocean, actually. And so that was at the same time that I was in the Canada Basin. Carlos and Ramon were doing that cruise just south of Svalbard. And I actually came here and was looking at that. And then later we did couple of overwintering expeditions. And the people here from Barcelona were also involved in those two expeditions, which were heavily led by University of Laval and myself and a lot of other people. So that's my history. Doop. And I used to be younger. I was looking for a picture of Carlos, and I only found this one on, which we've already seen before, but it's about that time. And so basically what happened was, as I mentioned, oh, it's this one, yeah. I was out here in the end of August 2002, and you guys were in here the end of August 2002. So suddenly we actually had DNA that had been filtered and collected within 3 days of each other on total opposite sides of the Arctic Ocean. Now, doesn't that sound like that ought to be a paper? I mean, let's do it. We got the whole Arctic Ocean here. And the first paper that came out of that was a nice, one of these grandiose titles where distribution of marine microbial eukaryotes in the Arctic Ocean and adjacent seas. I mean, that just covers it, eh? I think we did like 50 clone libraries. I'm not, that was back then. We couldn't do that now. Anyway, so the next thing that happened was out of that particular cruise, these are the clone, there must have been more than 50, yeah. I was in the lab constantly for a year and a half. And one of the ones we isolated, as I said, was this micromonas. We found my micromonas. It was kind of cool because you filter it down, you get DNA, you go over to the hospital, get it sequenced, and it comes back and it's a micromonas that matches one that I'd sequenced in previously that I'd isolated. And the other one I'm going to talk about today is this rhizarian here, which is also here. So these ones were actually isolated from the Canada Basin, basically, and these ones were from both sides of the Arctic, as I recall. And there was a lot of other cool things, including what we were calling pico-billy fights, which turns out to be not pico. Well, yes, pico, not billy, not fight. Anyway, but it worked. And the first author on that was Fabrice Knott, so we thought we should call it Knott Pico-billy Pico fights. But there. Anyway, that's enough of that. Let's go to the next slide. Why isn't this working? Okay, so then do a little bit of a discussion on mammaliales in the ocean, because there turns out there's more than, as Daniel said, there's also Bathycoccus up there. And this is a nice tree, basically giving you all you want to know about mammaliales, which was named after Irene Manton, who used to be called Mammy. Anyway, so you have the micromonas here, and then there's only 3 genera in this group. And then there's Osteococcus, which lives in warmer water, and Bathycoccus, which seems to be all over the place. Micromonas is all over the place, too. So when we first started this, there was only one described genus. And we kept working on this, and this is sort of a tree from August 2019, just looking at this whole cluster of mammaliales that we found from Nares Strait to the bottom of Baffin Bay. So one of the things that Carlos noticed during that Franklin Bay cases 2002, 2003 overwintering expedition was chlorophyll started coming up before the ice melted. And he sort of was on this thing. We really had to look at this and figure out where this chlorophyll was. And it turns out that it was chlorophyll B came up too. So chlorophyll B, that's in mammaliales, and there's also some other pigments that were in there that are only found in the mammaliales, presentophytes. Actually, there's one that's only found in micromonas. And that was in there. And so my husband, Warwick Vincent, had an HPLC machine. He kept telling me that we needed to do something with this. So between Warwick Vincent and Ramon and all these other people, and Carlos, we plotted out the Julian day, and sure enough, as the as the chlorophyll went up, from starting 20, this is like January 20th, if you remember your Julian days, you had this nice little thing in there, and it turns out that we also had some, microscope slides, and Ramon Tirado took this picture. He's a Barcelona boy, and actually I stole him from here. I took him and made him do a PhD with me in Quebec City, but so you could see that this was what's interesting. So at the time, keep in mind, everyone thought, okay, in the Arctic Ocean, freezes solid, absolutely nothing goes on. Everybody's just sort of sleeping like a polar bear no, but it turns out polar bears don't sleep. But they, just sort of like a brown bear nested up in its little cave, and it turns out it was really like a polar bear that was out wandering around looking to feed fur seals or something. So that was the first, and again, we were calling them picoprasinophytes at the time. And the Mammiales keeps moving in and out of the prasinophytes depending on who's doing the definition. So then the next overwintering expedition that the Amundsen, the ship that's based out of Quebec City, did was the Canadian Flaw Lead study, which was actually led by David Barber. And again, it was a thing where we put people on a ship. But this time, instead of being frozen solid and not moving more than 2 inches here in Franklin Bay, which is sort of here, that first overwintering, the ship would go up, and once a month it'd kind of run up this flaw lead, because where you've got the ice is moving this way on one side, and it's moving that way on the other side, and it makes what's called a flaw lead. And the ship was sort of going up and sampling that, and again, we got a nice seasonal signal out of it. And then my student, Natalie Jolly, who actually did a stage here with Ramon, I think. Maybe, I don't know if Carlos was here or not. She actually wrote this up finally with Ramiro Logares, who's also was here at the time. And so we looked at the seasonal patterns of this. And what's interesting is that if we just plot out the green stuff, things with chlorophyll B, there was, the Micromonas was here all the time. This is CCMP 2099, but there's also another one, which is Bravo. And this was the first time this was from massive sequencing. I think we were doing Illumina sequencing for this one. And the Bravo, which is this slightly darker green, it was in there, but you can see there is it was always there, and the only time you have a kind of a spike right here where the Micromonas 2099, I'll call it, disappears, that's like the spring diatom bloom. And also the other green that came in was Paramamonas, which is a slightly bigger cell with 4 flagella. And then it was always a bit of Mantoniella around. And Bathycoccus was there all the time too, which was kind of interesting. Because what's this? I thought that Micromonas was the only green algae up there. It turns out that was the only one I picked up in my clone libraries, but yeah. So there was other stuff. And this is, Ramiro did a tree with a different gene, the PRP8, and you had the same sort of different clades coming out with the Micromonas, Bathycoccus and Micromonas, and there's so you could see it was the same sort of diversity that really well mirrored the 18S gene, which was kind of nice. So Micromonas hasn't gone away. It keeps turning up all the time. And every time I've sent somebody up there or been up there myself, my first master's student, Marianne Potvin, she did a coastal and offshore study. And then the IPY was the Canada flaw lead study, which we just talked about. But then since then, you know, I've been working in the Canada Basin, North Water, where the original isolate was from, and all through the Canadian archipelago. And this, it's also, we've looked at Hudson Bay Complex, and then it keeps coming up from Fram Strait and Svalbard and East Green. And also on the Malena study, which was in 2008, I don't remember. No, it was after that. I was on a ship, I was on the Evanson with Danielle, and he isolated quite a few extra samples of this. And I remember at one time he told me it was really annoying because all he kept getting was micromonas, and he thought he was going to get lots and lots of phytoplankton. And so it's there, and it was Danielle's group. They formally described it in 2017 as Micromonas polaris. And then again, it keeps turning up in the green edge study, dark edge study, these things being run by Marcel Babin, who is also at University of Laval and has had well-funded big grants because he takes advantage of having a position in Europe, in France, and a position in Canada, and he has access to huge piles of money. So that was is that my Micromonas story? Oh yeah, no, my student Nastasia has been recently constructing mags from a transect that we've been doing for years between Ellesmere Island and Greenland. And she's managed to get 10 Micromonas, Bathycoccus mags, metagenome assembled genomes out of this, and this paper we're submitting, today, if I finish this talk. So basically, this is the nice part, as has been pointed out before, is once you start getting mags, you can start reconstructing entire metabolic pathways and biogeochemical cycles and get a really good idea of what's actually going on in the cell, but also you can look for those same genes and copies of those genes in other, just target those genes themselves that are of interest in different regions. Okay, and also Adam Monnier, who did a postdoc with me and is now a professor at Exeter, he went out on the Canada Basin and collected a lot of surface samples, and he's got a paper which should be submitted pretty soon, looking at the pan-Arctic Micromonas. So he's got cultures, the ones I isolated, plus he's pulled a lot out of the Roscoff culture collection, and mags, and is working on getting a pan-genome of Micromonas and how it adapts to the Arctic Ocean. And so this is, basically he sent me this picture, which is probably but you know, your postdoc goes and he takes, or that his student takes a picture, okay, we'll use that. And this it should be almost submitted. And so my argument here is that this Micromonas, it's just as endemic to the Arctic and just as characteristic as a polar bear. And so this thing, it gets on the cover of magazines and stuff like that. This one, especially if Adam's taking the picture, it's not going to make it. But anyway, this is what's nice. What started out as just sort of randomly culturing, as has been pointed out, and then looking at data and letting the data suggest what's going on here, exploratory, you know, basically no particular application science, you can actually keep exploiting that. And so this was where we go to the next part of the talk, I think, yeah, which is basically what I've been showing you so far is just, you know, water, but keep in mind that there's ice, and this is the seasonal, you know, basically ice follows this. And I put this on here because Josephine Rapp made this slide, and it's in a book by Paul Vossman now. And it just shows you, keep also reminding us that we have a surface mix layer, and it's a salinity stratified ocean, and that there are lots of different flagellates who are very diverse, as Danielle's pointed out, but there's also a lot of heterotrophic representatives. I mean, all of these people who study phytoplankton all the time, they always keep forgetting that half of that eukaryotic biomass in any ocean and they don't photosynthesize. They do other things. So this is the next part of my story. We're going to explore one of these ones that tends to be more heterotrophic. And this is just a quick reminder that ice is disappearing and stuff like that. I don't know why I put that in there. So what you need to be reminded of once more is that the Arctic is a semi-enclosed ocean. It's like a Mediterranean Sea because it's surrounded by the Mediterranean. And Nares Strait, which is here, is an outflow. So water, it goes around the Beaufort Gyre here and kind of comes through here. As I said earlier, Nares Strait is basically this comes through here, and this is northern Baffin Bay. It doesn't have any rivers going into it. So this is purely coming from either here or here. And then you've got this other current, the Pacific water, that comes in and comes through the archipelago and goes down. And so the big question, what happened here? I don't know. Oh, I hit the wrong button. Oops, this is what happens when someone's waving fingers at you. No. I did it wrong. Where am I? I want this one.
Which one?
I found this one here. Okay, yeah, so we went and we looked at Acantheria. And this got published in August. And again, it's by people with a history in Barcelona. And Carlos's legacy, including Mary Taylor, who did a stage here for 3 or 4 months, and Aurélie Labarre, who did her PhD with Ramon, and then did a postdoc with me. And so we looked at these Canada Basin samples, and also from the Nares Strait here. And Acantheria, if you don't know, what's kind of interesting about them is there's a couple of them that actually form cysts, and these are your, what do you call it? Poster child, poster protists from Haeckel, and the cysts can actually sink and form zoospores. And so we looked at a couple of clone libraries from the Canada Basin and Nares Strait, and this is that original graph from 2002 from that total Pan-Arctic biodiversity, and there is this clade B and a clade C of Acantheria that we found in there, according to the DeSalle classification. But these Acantheria, they were like 100% similar to, B was from the Northwind Ridge, which I'd isolated during that original study. And then this clade C stuff was what we isolated in Franklin Bay. Not isolated, cloned in Franklin Bay and looked at. So for this purposes, we call them the Northwind Ridge prototype and the Franklin Bay prototype. And then we sort of proved this by mapping them onto stuff. And they have different distributions. So the Franklin Bay one is pretty much just the Northwind Ridge one is just in the Canada Basin, which is here. These are all the big red dots. And the Franklin Bay one, it turns out it comes through Nares Strait. So it has to somehow get from this Pacific jet where we picked it up in Franklin Bay over the top of Canada and then down through here. And so basically what you do is you kind of look at maps of ice flows, which they do, and it must travel, I did it again. It must travel from the Canada Basin, which is here, and this is the speed. The blue stuff is going really fast. So this water, these ice flows kind of go along through here and then jet out. And the water does the same thing. So, and whereas all we got was this Franklin Bay clone in here pretty much. But this one we picked up in the Canada Basin, like at least every time we go up there in that part of the Canada Basin, it's there. And this actually points out there's uneven warming in the Arctic, so you have favorable conditions for Arctic and Atlantic species to come in. And there is now, I think this is the first time anyone's documented that you can have this northern route for organisms to get from the Pacific Ocean straight into the Atlantic, Baffin Bay, that part of the Arctic. And then this is my conclusion slides. Okay, we got Micromonas is incredible. There's two clades of Acantharia that offer lessons in generalizing too rapidly. One of these clades, it could have the one in the Northwind Wedge probably has photosymbionts in it. And Metagenomes is dishing up new insights. And this is basically, I'd like to thank all these people for everything. Thanks a lot. Oh, yeah.
Okay, thanks.
That's it.
Thanks, Honey, for the talk and for this visit to the Arctic. So now we are going to move on. We are going to change gears, no? I think this is how people said. Now we are going to leave a little bit these scientific talks. I'm moving to a more divertimento talk to cover one of the passions from Carlos, which is bird watching. I know that is a passion that is not only from Carlos, other people have this passion. And Klaus, that comes from the Institute in Warnemünde, marine research. He's an expert in anoxic environments, but today he will explain about bird watching.
You have to start here if you want to use that. It takes a while to. Oh, here.
Okay.
Ah, here. You have it.
And this is for pushing here, probably.
Yeah.
Okay. Yeah, first, thanks a lot, Ramon and Pep, for inviting me. It's a pleasure to be here in the Institute after a long time of absence, I must say. Well, a voyage with Carlos Pedrós-Alió, I also try to recall a few events, what I remember from my side. And I think really 1988, really long time ago, was my first visit to Barcelona after I had just finished my studies at the University of Constance. And I got some contact to Carlos via Hans Güde. And I gave a seminar about my diploma thesis at the Universidad Autónoma, where Carlos, Pep, and Ramon were still there as limnologists. And we even submitted a proposal to the EU, because I had the idea to join Carlos' group. But this proposal failed, and I started my PhD then in Plön, at the Max Planck in Germany. And it took a few more years for my next visit. That was I think it was in '92, shortly after the Olympics here in Barcelona. And Carlos, Pep, and Ramon had meanwhile moved to the Marine Institute. And it was the old institute, which was, well, it was really a nice atmosphere, fantastic people, but the labs and the cooling rooms, I still remember, was a little bit shocking for me when coming from the Max Planck. But on the other hand, there were these huge turtles in the basement, and this compensates for a lot, I think. And well, we did a field study, because I think they were still by heart limnologists at this time. They haven't arrived yet at the marine science community. So we went to Lake Siso to study these famous meromictic communities. It's really interesting for microbiology. But there was a Daphnia invasion at that time that has more or less exploited all these communities. Really, that was. But we nevertheless did some studies. Later on, I went on the Latitude cruise, thanks to Pep and Dolores. And I think in the 90s, we had really a lot of collaborations between the Max Planck and the ICM. Ramon, Montse were there for a longer time, and Pep and Dolores, I think, also. So unfortunately, I have no pictures from that time. I think we didn't have mobile or couldn't afford a camera, I don't know.
No more pictures. Well, then in 2003, I moved to the IOW in Warnemünde, so also my transition to the marine world was less time as a department, had less visits, but still some collaborations with Ramon, for example, were ongoing. So it was, it was an important time. 2018, Carlos was the last time at our institute and gave a talk, and we went for bird watching afterwards. So it's one of our common passions. And when in all this contact with Carlos and what developed from it was really important for me, not only scientifically, but also in terms of combining work and friendships and having fun, and of course these bird watching trips. And well, even though our paths no longer cross so often at conferences or so, I mean, this is a time I don't forget. Well, and Ramon and Pep asked me to give a talk about birds. A little bit astonishing, because I don't have a talk about birds. Maybe because they think I am scientifically not so active anymore, and I should talk about my passion, I don't know. But then I thought, well, in the latest cruises, I got more interested in seabirds, and maybe I can show some pictures from some last cruises. Also, it's to relax, no trees, no papers, etc. But a little bit of background to my field and to the cruises, because in the last 20 years or so, I work on oxygen minimum zones. First, of course, in the Baltic, in the basins of the Baltic that are anoxic. But as a comparative system, the Black Sea, which is in many aspects very similar. And in the last years, we moved on. You see in this map these huge oxygen minimum zones in the Pacific and also in the Atlantic. Well, we didn't visit the Indian Ocean, but we went to the Namibia upwelling in the associated oxygen minimum zone, and also to the ETSP, Eastern Tropical South Pacific oxygen minimum in front of Chile. And that's where some of the pictures come from. And just also as a background, what we are interested in studying biogeochemical processes across oxic-anoxic interfaces, across oxyclines, nitrogen processes, who is responsible for the nitrogen removal from the system, who is responsible, and how much N2O, nitrous oxide, as a climate-relevant gas is produced. In some system, in the sulfidic system, which organisms are responsible for detoxifying, for consuming the sulfide, so that it does not enter in the upper water layers. And it's interesting to compare different systems with different chemical profiles. The oxinic systems, where you have a sulfide nitrate or sulfide oxygen transition, or in the Black Sea, even more interesting, where you have no connection between the sulfidic and the oxic zone, and the question remains, what is oxidizing the sulfide? So we compare different systems, and of course also these Eastern Tropical South Pacific, where you have a midwater anoxy, or the Namibian shelf, where you have anoxic waters on the shelf. And one approach we do, before I come to the birds, I also want to just mention what we are doing. We do sample a lot of data for metatranscriptomics, and we do that by in situ fixation, because mRNA is degraded very quickly when you bring samples on board. So we fix the samples in situ in the respective water depths. And with a fixative, these are you see here these syringes. It's a relatively primitive system, but it works very well because the syringes are released at the respective water depths. And then we get the metatranscriptome, and from the parallel bottles, we get all other kind of things that we need. And we link normally these metatranscriptomic data to high-resolution chemical profiles that are taken in parallel with a pump CTD that is lowered in the water column, and that pumps water to the ship, to the lab, to a series of sensors for gas measurements, and to an autoanalyzer for nutrient measurements. And then we got high-resolution physicochemical profiles. This is, I think, very important in these systems where you have these strong gradients, and then you can have such a profile as this one, an example from the Black Sea, where you see this depth where you have the nitrite peak, where you really know then water is getting anoxic, or where you have a phosphate minimum. That's a story for itself, why there is a phosphate minimum. Organisms are involved. You can read that in this paper here from Jules Vogt, polyp magnetotactic bacteria, and also one example where we studied the sulfite oxidizers. But I don't go too much in detail to that. Coming to the Namibia cruise, Namibia upwelling and the Namibian shelf is interesting because from time to time you have these sulfite eruptions that you can see from in satellite pictures. When this happens, then people can collect the lobsters from the beach because all the animals come out of the water. And so we had this cruise in 2022 to the Namibian side. Here you see these transects we had on the shelf, different transects. Here's the port of Walfish Bay where we started, and here in blue is the temperature map. In blue you see clear signs of the upwelling. So we had strong upwelling in the austral summer. And we had also here a nice, for us nice, a nice oxygen minimum zone, but in the bottom waters. And just one last picture from the science part. Here these high-resolution profiles with a pump CTD. Indeed, we have here sulfidic bottom water and oxygen depletion, and that is something we are currently studying, the metatranscriptomic data. So the question is how are these profiles reflected in gene expression and hence in functions of microbial key players? Also something we do mainly for prokaryotes, but maybe in the future we do it with Ramon and Aleix also for looking for eukaryotes. What is the activity of eukaryotes in these profiles? Okay, but when not being a chief scientist, one has sufficient time in between sampling for bird watching and photography. And I had quite some time on this cruise. And so we start to look for coastal birds when we are in the harbor. And we see seagulls that look very similar to our seagulls here in Europe, but of course they are different species like the Cape gull or the hardlob's gull. And here flying over a great white pelican from the nearest estuary. And but there are also more interesting ones, endemic species for these West African or Southern African regions, such as the Cape cormorant, the Cape gannet, or the African penguin. And these are species that have been declining a lot already in the late 90s in parallel to the collapse in the sardine and anchovy fisheries. Also these birds declined a lot, and some of the colonies are also endangered now. Well, moving more outside from the coast, one sees also, there are still birds, but they look different. Here we were setting just a mooring here in more offshore, and you already see it. Birds are flying around, and these birds are obviously not seagulls. They look very differently. And they belong to this family of so-called tube-nosed seabirds. There are different families from the large albatrosses, storm petrels, to this diverse group of petrels, shearwaters, diving petrels. They all have these characteristic tubular nasal passage. That's for salt excretion, but of course also for olfaction. They have the ability for olfaction, for example, to smell DMS, and thereby to locate patchy food distribution in the sea. They have a cosmopolitan distribution all over the oceans. They breed mostly in remote islands, some in offshore islands, and they are also make many long-distance migrants all over the oceans. And some of them we could identify here on this cruise in front of Namibia. This was the most common one, this white-chinned petrel with this peculiar beak, which breeds in subantarctic islands, but then flies around all over the oceans. A spectacled petrel, a little bit less common, that breeds in this remote island, Tristan da Cunha, but also can be observed then when you go out with a ship. Another one, Cory's shearwater, that can be seen also in Europe here from time to time, but also everywhere in the ocean, or the sooty shearwater, a common one, another one that occurs in all oceans, can be also observed in Europe, but breeds, has large colonies in southern Chile and in New Zealand. And well, when looking at these assemblages, I have to see, now I'm totally wrong. How can I go back? Okay, but you see maybe better with this video. There are not only these shearwaters, but you already see there are some larger birds, and of course you know these are the albatrosses, so they also gather together with this, and this very close to the ship. And so a couple of albatross species were also present, like the white-capped albatross, also amazing distance, flying all over the southern, in the Southern Ocean or the Southern Atlantic, breeding near New Zealand, but then migrating here up to these Namibian waters. The Atlantic yellow-nosed albatross, another one coming from the other side, also breeding on Tristan da Cunha and then migrating towards Namibia, also a very nice bird. And the black-browed albatross, maybe many of you might have seen him, because it occurs in many, most over the oceans, breeds also on many subantarctic islands. And also here you can see this, get an impression of the quite numerous albatrosses that are gathering around the ship. A lot of activities, and well, but there are also some. The smallest of this family of tube-nosed birds are these storm petrels. This pretty tiny bird. It can really walk on the water, picking up small food items from the sea surface. It also breeds here in these subantarctic and Antarctic islands and can be observed there. And I mean, I was really wondering why there were these huge concentrations of seabirds at some spots, whereas then you drive for miles and miles and you hardly see any bird. And that could be a future project, combining what is under the water and what is above the water. And one indication might be here these measurements from our physicists. They measure the turbulence with a microstructure profiler in the water. And you see here at the edge shelf, we have really increased turbulence, which brings also potential food items to the surface. Again, birds are smelling or seeing these food items together, detritus, small fish, etc. And that might be one reason for these large concentrations of seabirds at certain spots outside. Whereas normally you would expect them after the fish trawlers, but obviously there were hardly any seabirds following the fish trawlers, maybe because the catches are meanwhile so low, I have no idea. The other interesting thing for birds there is that some of the birds we know from the northern hemisphere are staying over winter in the southern hemisphere, and we meet them. We might have seen them in the north, like this one here, this seagull. Sharon might know it. It's a Sabine gull, which you can sometimes see in the North Sea, which breeds in the Arctic, in the Canadian Arctic, but also in the Russian Arctic. And you see here it overwinters in front of South America and in front of South West Africa. So also really nice to see again these birds overwintering in the south. Also the European storm petrel belongs to that, that is breeding here in the coast and islands in Europe and overwinter or staying outside the breeding system, breeding season all over the ocean. And some of these so-called predatory gulls, jägers and skuas, they are also abundant there. They come also from the Arctic and spend the winter there, like this Pomarine jäger. And here you see the distribution of these birds. Normally you see the breeding season in the Arctic, and then when you make a cruise there in austral summer, you see them around the ship. There's also one species of these jägers that is resident there, that is not migrating. There's the skua, the subantarctic marine skua, a very aggressive predator and chasing other birds, like this large or similar sized Cape gull here. They can also be observed around about. Yeah, that's the impression from the birds from Namibia. And 1 minute, that's good, because now we begin with Chile, and I would need 20 minutes more, because that's the last picture from Namibia, and you see the Namibian desert there, and then moving to Chile, but I realized that time is not enough for Chile, where we did last year the cruise with a Sonne in the Chile upwelling, and that's only 2 pictures of 2 birds, because they are very nice birds in Chile. You might know this Inca tern that you can see already in the harbor and leaving. And when you go offshore, magnificent, really huge birds, the largest albatross we have, the wandering albatross you see here, and also the largest petrel we can see, so also fantastic birds going offshore from Chile. So yeah, thanks for your attention. I hope that you liked the pictures and that get some motivation to watch out for marine life on your next cruise. Thanks.
Thanks, Klaus, for this visit to the birds from the cruises. Now we move on. We are still...
Sorry, we are the rare biosphere here, and we made our presentations in keynote.
So, but Vanessa and Marta will talk about a little bit how science and art can find a connection. And I said before that was one of the, also of the interest of Carlos some decades ago. So Vanessa is a technician here. She's in charge of the molecular lab. And Marta Arroyo did the PhD recently with Sylvia.
Okay, so first of all, thanks, Pep, Ramon, for organizing this meeting to celebrate together Carles' legacy. And okay, so Marta was the last Carles PhD student here in the Institute, and I was his first technician. So Carles joined us, but also microbes, art, and science do. And I'm going to give a general introduction, and Marta then will tell you about a project that we were developing also with Pablo Sanchez, who is here. Okay. Okay. So when I started thinking which was my own journey with Carles, I realized that was one of constant learning. When I arrived here 24 years ago, so not bad, I was more interested in genetics than in microbes, but Carles shifted this perspective because I started understanding that the work I was doing in the lab could be connected with universal questions like how the world works or origins and limits of life, and I love that. Then I have the opportunity to share an Antarctic cruise with him, and from that experience I learned the beauty of observing nature and also the value of being patient, even cold weather. And another lesson he taught me was how important it is to share knowledge with society. And Carles' approach for that has been mainly through books Here you have a list of the books he has published, and Pep kindly displayed them outside, right? So you can have a look on them. Then another lesson I got from him was that I could connect our passions with scientific work. And here you have an example of a paper we wrote with Andrés Barbosa, linking birds and microbes. And then he did a super nice talk about art on science that had a deep impact on me, because then I started dreaming that I could connect contemporary art, which I love it, with the science that we were doing here in the Institute. And in that talk, sorry, Carlos, because I summarized in 2 minutes your 1 hour talk, he pointed 5 proposals where art and science could converge. The first one was that art and science arise from the same creative act, and for that he based it on a book of the 60s, I think, no? Yeah. That said that it is at the point where art and science converge where the creative act occurs. The second one was that art and science are biological activities that emerge from our primate brain, and for that he gave some examples, among others the aesthetic instinct of Congo, a chimpanzee that was considered painter and artist, and also the universality of the prehistoric paintings. The third proposal was that work of art and science need a context and an effort to be understood and enjoyed, and for that he compared the abstraction and simplicity with which Miro or Einstein could express the complexity of their work. In the fourth one, art and science use the same methodologies. He compared again all the research and experimentation that is behind this cubist painting by Picasso and all the effort that led the discovery of the DNA structure. And finally, they share the objective of discovering and communicating truths about the world around us. And for that, he went deeper into Dalí's work and how he connected it with the science of his time. And here I have listed some other thoughts and ideas about art and science. I love this one very much. They are the questions and not the protocols that make science and art. So from this first talk in the early 2000s, we had a very big gap in our institute until 2017, where we did our first collaboration with an artist, institutional collaboration. She was Ana Rierola, and she created ancestral forests using microscope images from our institute and talking about the importance of marine microbes in the world functioning. And then last year, we created a working group supported by our director on art and science, and we start establishing contacts with museums, with art and science foundations, just to find synergies and ways to collaborate with. And this year, we have written our Art and Sea program in the context of the new Severo Ochoa proposal and the project Barcelona Mar de Ciència. And just I would like to mention the European art and science projects we are involved in. The two first ones, Art for Sea and Tidal Arts, are about doing artistic residences in marine topics. And the last one, coordinated by La Fura dels Baus, was about training and challenging a group of artists that finally created a performance talking about currents, about plankton, and about microplastics. And now I will go very quickly on that. I just want to give you some examples on how we are working with contemporary artists now here in the Institute. She's Mafe Moscoso. She's an artist, an anthropologist, and she wanted to explore how scientists listen to the ocean to produce scientific knowledge and compare with other ways of listening that can produce, for example, traditional or indigenous knowledge. And now she's writing a book on her findings. Silvia Zaya was interested in exploring the anthropogenic noise and how it affects marine ecosystem. And he took torpedo rai as a model of perception, and he created a film that was premiered at CCCB. Here we collaborate with two dense companies, giving insights on plankton species, how they move, how they are, the roles that they play in the ecosystem, and things like that. And Big Bouncers created a dance show that was premiered at Mercat de les Flors and then in Cosmocaixa. And in the other one, Compañía Inervo, they are doing art pieces that will be exhibited next November in Louvre Festival. Here, with Elisabetta Broglio, we curated our first Art and Sea exhibition. It was a collective one, and we did it during the Ocean Decade conference. And here we have the opportunity to show two ongoing projects. The first one, again, by Ana Rierola. She's now focusing on ocean color satellite images and all the science behind these images, again, focusing on the phytoplankton roles. And she make these images dialogue with universal literature through a series of pantones. And here, Xavi Bou moved from his first previous work about birds' flight patterns, and now he moves to the microscopic world, and he is developing some gadgets to be displayed on our microscope so he can do chronophotography on them. And this one is Pep Vidal. He's a mathematic, an artist, and he did an exhibition where he wanted to explore all the science that is behind more than 100 paintings done by Cézanne about the Mont Saint-Victoire, which is at 1,011 meters of altitude, and all the science that is behind this bottle of seawater that we took from him in our last cruise in the Atlantic Ocean at 1,011 meters depth. And all the exhibition was an open reflection on how we build knowledge by observing nature. And here was an experiment too. It was the first time that we explained an oceanographic cruise by collaborating with artists and with the students of fine arts and design. And here you have some of the results, and you can check if you want. You can check them in our webpage, ICM Divulga, Expedicions, Mi Color 2, Correspondències Aranci. And finally, this is the last one. We are now working with Bau University and CCCB in a new project. And our starting point is Agnès Bardà Film, The Gleaners. And what we want to do is to glean on scientific data that have been taken for the projects, but have been not used for scientific proposals, like writing a paper, because they are unfocused images, contaminated samples, unknown species, unknown signals. And we want to give them to a group of artists so they can reinterpret them. And we are not interested in the final artistical production, but in the process that can take place to understand more how we can dialogue artists and scientists. And that's all for my part. So now Marta.
Hi, everybody. It feels so good to be back home, especially for such a special occasion, and even more so because I'm given the opportunity to talk about a project that links very well with Carlos' legacy, which goes beyond scientific research and develops new methodologies of communicating and transmitting just how the world works, thinking about other audiences and new formats. So Telestasi is an art and science project that was funded by the Catalan Foundation for Research and Innovation. And this is the team that participated in developing this project. It includes a wide range of profiles that go from designers, artists, creative coders, creative technologists, biologists, microbial ecologists, and bioinformaticians. So this is more or less how the team is composed. We have a scientific team from the ICM that is Vanessa Balaguer and Pablo Sanchez. We have two artists that are Alba G. Corral and Santi Villanova. They are digital artists and use generative coding to create visuals and sound design from algorithms that they can feed with data sets. Sometimes these data sets are random, and we challenge them to use real data from the ocean. And in between both disciplines, there's Tony and myself, and we have this hybrid profile, and our role here was to mediate this relationship and see how we could create a common knowledge to understand what goes on in both sides of the story. So the aim of the project was to communicate two simple messages that are very ingrained in our microbial ecologists' minds, but the common people do not know. One of these is that there's a huge amount of invisible microorganisms in the ocean, and they have an amazing role in allowing this inorganic chemistry of our planet to get into life, basically. So they link the ocean with the atmosphere and participate in climate regulation processes. So this was kind of a way to make people aware that changes in this microorganisms environment really has global consequences. So in true fashion of the research that takes place here, we challenge these artists into using these metagenomic data sets that throughout the years we've been collecting to talk about, in their own language, about processes of climate regulation, the albedo effect, deep sea carbon sequestration, and so on. So the first challenge of this project was to find a narrative, because you can imagine these are very complex concepts. And we did a super cool session on talking about microbial ecology, microorganisms, and life on the planet from a wide range of perspectives that went from ecology, biogeochemistry, genetics, the role of microbes in the present, estimations for the future, the origins of life on Earth. And not only from this scientific perspective, but also from philosophical perspective. We talked about Greek mythology, how we perceived the ocean through different languages that could be sound, Greek mythology, sound design, navigation, data visualization, and so on. So this kind of set a path in which we realized that we were actually talking about balance in the end. So that's what inspired the name of the project, this biogeochemical balance that allows us basically to be alive in this planet. So again, a very complex topic. We thought that the best way for people to understand like global warming processes and climate change was to actually be aware of how these microorganisms participate in processes that regulate the atmosphere's temperature, which is, in our case, we selected two processes. The first was cloud formation, so albedo effect, and the second one was this particle sinking and deep carbon sequestration. Next step was to find a data set, and after comparing all the different variables and samples that the department had collected through the years, we found that these Malaspina deep vertical profiles covered a very huge geographical gradient and also a super interesting vertical gradient for our purpose. So we thought that it would be interesting for you to know how this process was developed. And first of all, as scientists, we created a GitLab repository where we could have all the data, the code, and the documentation super traceable for artists and scientists. And I thought I had more slides about this, but basically, an interesting part of this is that we had the data by scientist folder and the data for artist folders. That means a lot in terms of, we think that these creative coders and these generative artists work with data, so our raw data sets will work fine, maybe separate tables in between taxonomy, functional annotation, and so on. But really, it took us a lot of work into understanding the kind of data that the artists needed and to transform our data sets into something that could be actually useful for them, considering that they don't speak microbial ecology. So it took us a lot of hard work in developing educational resources for them, explaining concepts that are super simple for us but not so intuitive for other people, like the difference between a station or a sample, why we have specific samples for biological metagenomic data, for example, but the CDD gives us a vertical profile with a lot of resolution and continuous data. We talked a lot, many questions arose about how this water column, what were the characteristics of the water column, what were the biogeochemical processes involved in the messages that we wanted to convey, which variables could be interesting to visualize somehow together because they made sense scientifically, and other concepts like what is a taxonomical classification, what are the different levels, and why does it matter? Another step, a very important step for Alba, for example, which is a visual artist, was to have a visual language. A common language that we shared was how do we imagine and what we know from scientific records, the look of microbes, the aggregates, and their dynamics. She really dug in the science. The first thing she did without us telling her was downloading Ocean Data View, and she started plotting variables, asking a lot of questions, and it was super nice to see her interest in really understanding the processes that she would later translate visually from an abstract perspective. These are some of her visualizations with Ocean Data View. She coded in Python, so these are plots from her Jupyter notebook. And on the other side, we also played with scientists to get them in the mood of, okay, we're producing a creative project, an art piece. Let's look at the different audiences that will likely visit our piece. What do they know? What do we want them to end up knowing? How do we think they will feel? How we want them to feel when they leave? And with all that, we were able to create this storyboard of this vertical journey that is the artistic piece, in which we talked about the different scientific processes that happened in the different layers, how our emotional take on the different biogeochemical processes, as weird as this may sound, the data variables that could be associated to each of these layers, and visual references and sound design. So in the end, Alba proposed this visual sequence that would be representative of all these layers, and Santi designed some algorithms which he plugged data to create the sound for each layer. I wanted to show you because these videos are super illustrative of his work. So he keeps tuning in different variables and creating soundscapes by adding different layers that could mean maybe chlorophyll, oxygen, temperature gradients, and so on. And this would be like the sample's soundscape. And he also added like a more romantic layer to that by converting ACGT, the nucleotides, into different nodes. Here's a little video of him explaining.
Hola. Pues resulta que, no, començant a pensar en els temes genètics, no, GATC, G en la notació anglesa és sol, A en la notació anglesa és la, T no és res, però com que no és res tinc llibertat, doncs aquesta, que és si bemoll, i C és do en la notació anglesa. Vale? Llavors, si improvisant una mica, no, sobre aquestes quatre notes, surten coses com...
Sorry, you will not be able to see it finish, but I don't have time. So basically, this is the vertical trip of the art piece. We will show you a teaser in a minute. We start at the interface. Here, we wanted to convey this gas exchange by sound. Then we take a dip into the surface waters. We compare different samples, one which is more nutrient-rich, other that is more oligotrophic. Organic matter starts being exuded. It falls into dim light layers. These particles get degraded. Some samples have certain metabolisms that others don't. This marine snow gets degraded. Bacteria switch from free-living to particle-attached. Then we get to the bathypelagic, where despite maybe environmental physicochemical parameters start getting more constant, there's a burst in microbiological diversity and functional diversity. And we reach the bathypelagic layer with a lot of different metabolisms until we wrap all the project up with this like kind of Gaia feeling. And we presented this project at Euphonic Festival last summer. It was on for 3 days, and around 240 people got to visit the piece. And we have been, throughout the year, breaking boundaries and transferring microbial ecology and its link to art in different cultural scenarios. That would be the Design Hub, which is the Barcelona's Design Museum, the Sonar Festival, and also the Euphonic Festival. And here you have the Instagram, so you can follow us, and a little booklet that we did as an educational resource for you to download. Do I have 1 minute for the teaser of, it's one.
A ver, Pepa me está diciendo algo por interno. Te vemos y te oímos. Ya, entonces empiezo. Ya, ok. Bueno, I will switch to English then, even though I will prefer to speak a little bit before in Spanish, just to be like in the Oscar and say, Carlos! Because you are really the winner, my dear. And today, we are so honored, all of us, to be here, and me in particular, and I really thank Pep and Ramon for this invitation.
Carlos is just move a moment to the bathroom, so I think if
The
you are so much enthusiastic, perhaps we should wait for him. And now he's entering, so please start again.
Okay, I will start again. Hola, hola, ¿cómo están todos? Hello, everybody. How are you doing? Thank you very much, Pep and Ramon, for this invitation. I think today is a very special day for all of us, and it's an honor to be here. And again, I will say, as in the Oscars, I have to say, Carlos! Because this is a very special day, and this is your day, and I think you deserve to be called for your name loudly, because I was expecting for this so many years now, and finally, Ramon and Pep, and thank you very much again to you too, to organize this seminar and give us all the opportunity to say thank you very much, Carlos, for everything you did. And it's a truly shame for me not to be there in person, but at least I can express my gratitude from all distance for all the science and experience that I shared with you, Carlos, and with all of you long time ago for some of you. And I don't gonna talk today. It was difficult for me to decide what to talk about. And even though Pep and Carlos pushed me a little bit to do that about extremophiles, actually, I think that's the right thing to do, because while I could have chosen many different topics, because Carlos has been there since the very beginning of my scientific life, today I select microbial ecology of thermophiles because I believe this is where Carlos has had the most impact on my research since becoming an independent researcher. So also it was with this scientific theme that I first arrived to Chile, and today is gonna be very much about that. And for nearly 15 years now, I can't believe that, Carlos has supported and encouraged this research that I have been doing in my lab here in Chile. And this is basically because Carlos is passionate about nature, particularly about extreme environments and the organisms that inhabit them, from microbes to plants and animals too. And this has been reflected not only in the research that he has been published in many numerous papers and very interesting and incredible papers, research papers, but also in his popular scientific books, where there is always a place for Chile and its biodiversity. So thank you very much again, Carlos, for including Chile and the work that many of us we are doing there, and or here. And just pull out the biodiversity that we have in this country. And as I mentioned before, it was through him that I got touch with some people in Chile. Monica, I know that you are there connected somewhere. And he passed on his fascination for Chile to me, convincing me that moving to the southern hemisphere across ocean was a fantastic idea. So since then, Chile and its microbial biodiversity has become my scientific world and a paradise for studying microbial ecology on thermophiles. Because if there is one extreme environment in Chile that is rich in, there are hot springs. And that is due to that Chile being part of the Pacific Ring of Fire in the American continent, where active volcanism generates multiple hot springs and geysers scattered across the country from north to south. For example, in El Tatio, in the north part of Chile, in the Atacama Desert, this is the third geothermal field in extension in the world and the largest in Latin America. So it's a very important group of hot springs located in the north of Chile, very interesting ones, and then I'm going to show you some results in a minute. But also, in more or less in the north of Patagonia, we also have other hot springs that have been the focus of many of my research together with Carlos in Chile. So, and it is there in these terrestrial hot spring environments with wide physicochemical gradients over short distances that make them ideal highlands models for studying species distribution and diversity. We can identify abiotic and biotic interactions in a large and small scales in these hot springs, also study microbial biogeography and endemism patterns, and assess role of these thermophiles in biochemical cycles to evaluate finally their potential adaptations and those that probably do make them evolve under these extreme conditions. In my lab, then since I got to Chile, I always, under the attentive guidance and collaboration with Carlos, we have worked to understand these microbial communities, these thermophiles, at different scales from local to global. And just like in hot springs of Patagonia, where Carlos took part in several of the sampling expeditions that we did in this hot spring, given his all along the way and enjoying this wonderful region of Chile together with me and many of you that also have been there working with me. And thank you very much to all of you to support my research here. And Carlos' ideas and contribution during the first project that we have funded by the Spanish government, Thermo Chile, made me fall in love with these ecosystems and leave me with a legacy of what will become more than 14 years studying thermophiles. In particular, the study of our model hot spring for many years, Porcelana, which has like this exuberant photoautotrophic mats growing abundantly, which has offered us the opportunity to investigate microbial consortia along the thermal gradient. These self-sustained benthic ecosystems comprising the main element cycles, trophic levels, and food webs, allowing us to study taxonomic changes, metabolic processes, and interactions along the temperature gradient. Thanks to the Chilean project DiasoSpring, then in my lab, once 1 year passed since I moved here, we were able to explore and rebuild using different methodologies such as stable isotopes to obtain rates, assimilation rates, and enzymatic activities and gene expression, using them both in situ and in vitro to get into the carbon and nitrogen fixation and assimilation across temperature gradients in these hot springs like Porcelana. Further, this project also provides us with the opportunity to employ metagenomic techniques to gain a deeper understanding of the main players in these significant processes related to carbon and nitrogen cycles through their functional capacities and also transcriptional activities. This allows us not only to identify the specific organisms involved in different pathways in the carbon and nitrogen cycles, but also to understand their preference, niches, and temperature dependencies. And if it is precisely in this way that one of these key players come along, the thermophilic filamentous cyanobacteria called Fischerella, the genus Fischerella, and particularly the species Fisharella thermalis, that become one of our model organisms in my lab to study their distribution, contribution, and adaptations along the thermal gradient in environments like Porcelana. For instance, regarding adaptation to temperature at the proteomic level, our work demonstrated that the existence of distinct ecotypes along the thermal gradient, both associated to photosynthesis and nitrogen fixation, very important processes in this cyanobacteria. There was another project called Metaspring, where we could also try to understand a little bit more about the effect of abiotic factors and geographical distance on microbial communities in hot springs in a more global scale. And Carlos was also involved in this project. And with this, we, for instance, understand more about the global diversity based on 16S rRNA sequences from more than 200 samples across hot spring of 4 different continents, that compare hot spring in Chile, like El Tatio, such as those located in New Zealand, Yellowstone, in United States, and China. This study demonstrates that there is no global model, like using, for instance, geographical distance as a parameter that explains better the beta diversity behavior in these thermophiles. However, local models prove to be more explanatory using physicochemical parameters like temperature, pH, or sulfates, for instance, that were more relevant in a local scale. So that was very interesting to study and to prove. Also on a global scale, is that we also investigate taxonomic metabolic novelty through the recovery of more than 4,000 thermophilic prokaryotic MAGs assembled from metagenomics and found in different databases and our own metagenomes. And that revealed some niche preferences and significant species-level novelty, which demonstrate a high contribution of these new MAGs to poorly described phyla that have low number of genomes present in the genomic taxonomic database, the GTDB. So that was very important. And some of these were cyanobacteria, which allow us to try to understand more about this thermophilic group of cyanobacteria, compare basically with the non-thermal cyanobacteria relative. For these genomic comparisons, we demonstrate that there is a variation such as smaller genomes and with higher GC content, as well as shorter and more hydrophobic proteins, and then also a more frequent number of appearance of CRISPR-Cas defense system, which has been talked in literature that thermophiles or species that live at high temperature, normally they have this type of defense systems more than others. And for that, that led us to, with the idea that we should explore more in detail the diversity of these defense systems in thermophiles worldwide, using many metagenomes, as I mentioned before, and using the Cas1 protein as a marker gene. Or as a marking protein. Our findings, or mere findings, reveal a specific diversity associated with geographic origin, which suggests the existence of a potential endemism between phage-host pair interactions. Given that, or those results, is that we realize that our future studies should focus on the biotic relationships between these phages and their potential host. To achieve this, It was essential to investigate viral communities in these thermophilic environments, which remain largely unexplored from a viral perspective from both at local and global scales. So thanks to that is that we managed to discover in Porcelana hot spring, our pet hot spring at that moment, the first thermophilic sign of age associated with one thermophilic cyanobacteria, in this case, Fischerella thermalis. And we could characterize that this interaction was related by a lytic interaction that over time stimulates probably the coevolution between these two pairs. Then we extend our studies on viral communities thanks to a new project that we got in Chile that was exclusively dedicated to study these thermophiles and particularly the interactions between phage or viruses and host partners and local adaptations, given that this geothermal field in El Tatio, which is the new pet in our lab, hot spring system, has unique features like geographical isolation, high altitude, high UV radiation, low precipitation, and this geographic isolation is very important in this area, among other features that make it a unique environment to study local adaptations. And this is how they look, the hot spring in El Tatio that we have been sampling for many years now in my lab. Here, in this El Tatio geothermal field, our main question to address was, can the geographic isolation and uniqueness of El Tatio select for endemic local viral communities? And that was very into the big questions that Carlos has always been talking about and inspiring us about our research. And some of our results first reveal that more than 80% of the viral operational taxonomic units, or viral OTUs, in El Tatio were unique compared to the RepSec global viral databases, as you can see here. And however, when we analyze the global viral protein cluster networks, we discover a significant taxonomic novelty among viral genera, where most of these viruses, including those from El Tatio, cluster always together among other hot spring sequences. That suggests that these viruses are very much restricted to these environments compared with the rest of viruses that are inhabiting in many other ecosystems all over the world. However, on the other hand, when we conducted beta diversity studies, we found, in turn, that these viral OTUs were distributed very much associated to a geographical specific origin or regions, which also suggests a limitation, global limitation for dispersion, but indicates that here we have local and regional adaptations that could happen in each one of these geothermal fields all over the world. Finally, from 2 years ago, I got a new project that is called Metamobilome, and here we keep working in El Tatio, our pet geothermal system from some years now, to explore and use it as a model ecosystem to study local mobile genetic elements, dispersal, prevalence, and role in host adaptation and eco-evolution. And it's in this new project that we step beyond of confines of cells and organisms, working from the air down to the microbial mats to identify mobile genetic elements that, through their dispersal, could influence genetic flow in these thermophilic consortia and provide more explanation for some of the remarkable adaptation and processes that thermophiles have in these amazing stream environments. So I just want to thank my talk Thank you very much, my people, former and present PhD and postdocs, as well as master students and undergraduate students who did amazing work for all these 14 years in my lab, and all my collaborators, many in Chile, like Diego Morata, Raquel Quattrini, and Monica Vázquez, and many others that have been working with me in many of my projects, not only in my thermophilic projects, also in my polar marine science projects, where Carles was also involved in many of them. And definitely my collaborators outside Chile, which have been really supporting, like many of you in Spain, like Pepa, Fernando, Francisco, Mojica, Martin outside, first in MIT and this later in Vienna, Ramón Rosselló, Javier Tamames, Virgitta Berman, Antonio Delgado, and many others that I'm sure they are there, and they have been supporting me in one way or another, or inspiring me in one way or another to keep doing my research outside my country, but still with a strong connection with all of you. And definitely, and of course, once again, thank you so much, Carlos, for all your support, wise advice. You have been a source of inspiration for all of us, and definitely for me since the beginning, and for all and each one of my projects. And I hope you will continue to be so in the coming years, during which I look forward to our continued collaboration, not only to uncover new knowledge, but also to enjoy this thermal and many other journeys together. So thank you so much, Carlos, and thank you all of you for your attention. And besos a todos. Gracias.
Thank you very much, Bea. That was a fantastic contribution from the Southern Hemisphere. We appreciate a lot that you were here, that you had to wake up very early to give this talk. Thank you very much. So, and now we move to the... What?
It was a pleasure. So thank you so much to give me the opportunity.
It was nice to
be
see you here,
today.
obviously. And so now we move to the last contribution of this session, which is Carlos himself.
Sense les ulleres no la veig. Meaning and sense o alguna cosa així. Sense. Well, as you can imagine, this event is very emotional for me. I'm deeply moved, but don't worry, I'm not going to cry. I think. I'm very thankful to Pep and Ramon for having thought about this and for organizing it. I'm very thankful to all of you for being here, especially those who came from outside Barcelona, very especially those who came from out of Spain, and very, very, very especially to that one who came from the other side of the Atlantic. I'm really thankful for that. The other question is to thank those of you who gave talks today, because they were not only excellent talks, but you attributed to me ideas that they were actually yours. So thanks a lot for that. And then let's start in search of meaning and sense, particularly the latter. One of the things one has to do in this kind of talk is to review his whole life. But since this would be very lengthy and very boring, I have decided to summarize my whole life in just one slide. So there it is. So now I can choose whatever I like out of this life and Won't be a problem. Yeah? The second thing is that one has to thank and name everybody, and this again is impossible. So I've chosen our favorite curve that has appeared already a couple of times, but in this case it's not a rank abundance curve. Certainly people are arranged in ranked here, but in this axis we have the importance of the relationship with me. Of course, a professional relationship, not just a personal relationship. And of course, you would organize people in this way, more or less, depending on the importance of the relationships, and everybody is in there. Still, I'm going to cite a few of those. Of course, mentors are the most important. They provide role models, they show us ways to do research, they show us the future we can have. And my two mentors are here, Ricard Guerrero and Tom Brock. Kathy is Tom's wife. And the two things that I most admire from them, and the ones I'm more thankful for, are first, the passion for knowledge. This curiosity to know how the world functions, this passion to know how things are. And the second one is that you have to finish things. So if you write a paper, you have to finish it, and you have to try to make it in the best possible way. So these two aspects, I think, are things that I've carried with me through the years. Okay, then, of course, you have the PhD students. The relationship with the PhD students is long and is intense, and it has a very nice feature. At the beginning, the student asks a lot of questions. If everything goes well, at the end, the student answers those questions. And I think this shift from asking to being able to answer is the keystone of a successful PhD thesis. Since I'm usually very lazy, I've had the help from many people to co-direct this thesis. In fact, many of those theses were directed by the other person, and I was just sort of hanging around. And then, of course, we have the wonderful technicians of the marine biology department. You don't know how much I have missed you these years. Those of you in the marine biology department should be aware that in these ladies you have a treasure, a precious treasure that not many other people have in many other labs. And then, of course, you have postdocs and you have colleagues and everybody. All of you are there somewhere. So you will have to forgive me if I don't mention you again. Okay, then let's go for the beginning. You might say, okay, maybe this is going too far back. But how about this? 6 years old. We lived in Barcelona, in Puigset, which is a little hill next to the Tibidabo, and the light contamination then in the city was not what it is today, so you could actually see the stars. So my father would take me to the roof of the house, and would point the North Star, the Big Dipper, the W of Cassiopeia, and all these stars. But one night, there was a little light that appeared in the western horizon, and it moved up among the stars and on the other side and disappeared on the other side. And my father bent down and whispered, Sputnik. Of course, at that moment, I wasn't aware of this, but it was witnessing the birth of a new era in which we live today. Nothing of what we do today would be understandable without the satellites. And I had the privilege to see one of the first ones going across the sky. The other thing is that realizing that something done by human beings could be up there with the stars meant that science was a very powerful tool to understand the world. And I think because of that, at least partially, I always knew I would go into science. Okay, that was my first enlightenment. The second enlightenment, here I was 17. I was beginning my studies at the university, and with my friend Ignacio Fita, we went to the Cadí. This is a mountain range parallel to the Pyrenees, and those of you who have been there know that the northern side is extremely vertical, vertical cliffs. And we were walking above, out here, and trying to traverse the whole thing. And then one evening, we saw this huge storm coming from the Pyrenees. There was this thunderstorm and lightning and clouds, very dark clouds, coming towards us. So we decided to take refuge in this little cave. This is a ledge, this is the cliff, there is a little hole here, and from the inside you can see this cave, and there's a little fountain. So we went there, and we remained there because there was nothing else we could do. So the storm came in full blast, and the whole mountain was vibrating, and it looked like something exceptional was going to happen. Actually, nothing exceptional happened, but both Ignacio and I had one book with us, precisely for these occasions, and he had, well, this is Storm. He had this book, Ficciones by Jorge Luis Borges, and meanwhile, I was there, I began to read it, and I've never been able to stop reading Spanish-American literature. This was one thing that was very nice. And I think I had this other book from Alan Watts. Alan Watts was not a master, he was a communicator, but for people in my generation, he was great in bringing to us knowledge about Eastern philosophies. And the main thing about this book was that I realized that religion and spirituality didn't need to go together. So you could have one or the other, or both, or none, but they didn't need to go together necessarily. So all these enlightenments, I decided to go to the United States to do my PhD. You've seen already this map. I don't know where did you see this Statue of Liberty thing. So I was really amazed that you had it there. And I worked in this lake. And Madison was an extremely nice city. It was a city that, even though it is in the US, I was able to live there for 4 years without a car. I could bike anywhere, which is something really unusual. And I learned many things. This was my department of bacteriology with a long tradition. And I learned, as I said, many things. For example, how to live with a cold, or how to work through the ice, which came in handy afterwards in the Arctic many years later. And I learned so many things and experienced so many things. For example, I met Christina, with whom I've made a team that is lasting over 40 years already. So everything was so important and so emotional that the only way I could deal with it was writing a fiction book. And I wrote El Diablo de Tasmania, Tasmanian Devil. This is a thriller, scientific thriller. There's a very ambitious professor in Madison, of course, who tries to synthesize life in the lab, and who is also in search of the fourth domain of life. So bacteria, archaea, eukarya, he's going to find the fourth one. And then two postdocs converge. One is a bioinformatician from Barcelona, a male, and the other is a molecular biologist from Colombia, a female. And so everything you can imagine happens. Samples disappear, there are murders, there are adventures in the Arctic and adventures in the Colombian rainforest, everything. So if you have been to the US for a few years, or if you're planning to go, I think you'll have fun reading this book. And the key scene actually happens in this called Science Hall in this place here. So you can identify what happens. Now, I was not able to publish this book. No publisher wanted to publish it. So I had to take it to Amazon. And I was very frustrated until I realized that this Nobel Prize winner, Joachim Frank, had the same problem that I had. In his webpage, he has this, wants to be a writer. And this is the beginning of one of his fiction books that he couldn't publish. So I was so amazed by this that I wrote a little piece for the blog at the CNB, mentioning how many scientists actually like to go into the humanities, into painting, into writing, into music, and everything like that. But it's very strange that somebody in the humanities will jump into sciences. And I just left us as a question, what does this mean? And he must have some kind of alarm that whenever anybody mentions his name in the internet, he gets a sign. So he wrote to me and said, "Dear Carlos, thanks for your generous posting." I don't know why he thinks it's generous, because it wasn't, but drawing attention to the fact that people now take for granted that scientists are summarily, to use a German expression, Fachidioten. So I said, okay, if a Nobel Prize winner cannot publish his book, of course, I cannot either. So I'm happy. However, very recently, he sent me another email Just let you know that my novel, Ia Petra, or His Sister's Keeper, has just appeared. So now I'm depressed again. For some reason, a lot of my career has been linked to the Americas. And I've had the need to write books about the things that I was seeing. So we have Desert of Water, about a cruise in the Arctic, where, for example, well, many of you were involved actually in that cruise. Then we have the Tasmanian Devil, about my time in Madison. Then we have Under the Ocean Skin, which is about life in the oceans. Then biodiversity, this was published just recently. A lot of it happens in Costa Rica, even though there are also parts in Canary Islands, in Galapagos and Hawaii. Then the plants of Atacama, and then the Life to the Limit. And this is my latest fiction book, El Río de las Vueltas. It happens in Patagonia. And again, I'm not being able to publish it, so we'll see what happens with it. Okay, for the rest of the talk, I'm going to touch 2 points, 2 lines of research that have been very important for me throughout my life. One is diversity of life, and the other, the limits of life. And diversity of life begins, for example, in this cloud forest in Costa Rica, where I was there with my guide, and at night, and he was looking for little frogs to show them to me. And he would find one, and this is so-and-so, and another one, and another one, and another one. And then I asked myself, why are there so many little frogs in Costa Rica? Well, there could be only one. The only function they perform is to eat arthropods and to be eaten by their predators. Why so many? Why not just one? The same thing, well, here you have the little frogs, which are really cute. But then the same thing happens with everything in biology. This is a partial sample of my collection of pine cones. Their function is just one, is growing the seeds, protecting them, and releasing them at the appropriate time. So they could be an optimal design. But no, there are many different designs. And the same thing for photosynthetic organisms. This map here shows the number of species of plants in different areas of the Earth. So, at least for land plants, for the photosynthesizers on land, we know how many species are there, more or less. And there are about 370,000. Really? Do we really need 370,000 species to do just photosynthesis? This is truly amazing. I find it awesome. I don't understand why this is the case. Of course, in the case of the oceans, we don't even know how many species we have there, but we know that mostly microorganisms. Many years ago, Ramon Margalef used this metaphor. Here we have the helmets of different firemen, and they have this ornamentation, which is completely useless for their function, but it's there. So we like to make variations of things. And he was wondering whether these helmets of different Daphnia species were also as useless as the helmets of the firemen. And of course they are not. In the case of the Daphnias, the helmets depend on the predators present, depending on the size of the mouth of the predator. So they try to avoid predation by having helmets. But then this gives a cue as to why there are so many species. There are always hidden functions, hidden subtleties that usually we're not aware of. But of course, if there are so many variations, then the next question is, how many species are there? How many living beings do we share the Earth with? And this is one estimate that I think some of you showed this morning. Maybe to you, Sylvia? I don't know. The laws say no? Well, somebody of you showed this estimate. And we have here is the abundance of organisms in one or in different ecosystems. And here we have the number of species in those ecosystems. And this relationship between number of individuals and number of species is one of those macroecological relationships which holds essentially everywhere. So if you extrapolate to the number of organisms in the world, which is 10 to the 30th, and then you see how many species you have, is 10 to the 12th. So it's 1 billion in the European sense, 1 million of million species. If this is true, I mean, we only have 1.8 million species described. So our ignorance is amazing, it's incredible. But you will say, okay, this is just one estimate and it's probably wrong. Okay, then I'll show you a different estimate done by Robert May many years ago. In this case, the macroecological relationship is between the size of the organism here and the number of species here. So there are very few species of the size of the elephant or the rhino, but there are more species you become smaller. For insects, we have about 1 million described, and then the relationship breaks down. Robert May said that it breaks down because our taxonomy of the smaller organisms is very imperfect. If we were to know them well, this relationship would keep going. So I asked myself, what's going to happen if I extrapolate this relationship to the size of bacteria? And this is what happens, 10 to the 12. Isn't that amazing? Two completely different ways of estimate, they come to the same estimate. Well, just amazing. Now, the thing is, these are estimates, so maybe this is not real. However, there are reasons to think that there are a lot of species of microorganisms. You have already shown this curve here, and the nice thing about this part here, the rare biosphere, is that you are protected from most death mechanisms. You are small and not growing, so you're not very nutritious, and predators don't like you. They prefer the bacteria which are growing actively. And on the other hand, viruses don't find you because you are too rare, and they need a substantial density of prey to be able to attack. So you can be there for very long periods of time. And then I realized another amazing thing, and that's that they actually can travel in time. Here you have the same core, and the organisms which are abundant here, every year they fall down on the polar cap in Antarctica with dust, with dry and wet deposition. They make a little layer, it freezes. Next year another layer comes, and another one, and another one. And this original layer keeps moving down the 4 kilometers of the polar cap, and then it moves out of Antarctica and breaks down in icebergs, and the icebergs melt, and they release happily the sample of the microbiota of millions of years ago. Then, of course, they become part of this rare biosphere, but if they find adequate conditions, they can grow back again and then be trapped in a period of ice ball, or they can be trapped in permafrost or in glaciers for thousands of years, or in sea ice for shorter periods of time. So potentially we could have with us bacteria almost from the origin of life. I realize this is crazy, and probably it's not true, but certainly you can have a lot of species from the very, very past, from the very far away in the past. So yes, there is a possibility that there are many different species up there, even though I don't know they are 20 to the 12. So if there are so many species, we should try to describe them. You know, this is something urgent. And for many years in my career, I was very frustrated because I had not described any single species. And then, thanks to Jaron Pinassi first, and then thanks to Sylvia and to Isabel Sanz, we could describe a few species, maybe 10 or 12 or something like that. So now I'm sufficiently happy. I'm particularly happy about this one because it's called Leeuwenhoekiella blandensis. Leeuwenhoekiella is the name of Leeuwenhoek, the founder or the discoverer of the microorganisms, and Blandis is the Roman name of Blanes, where people here at the Institute keep one of these microbial observatories. So I'm very happy about the name of this one. And lately, I've become interested in other things, and I have even done a little bit of work on plants. And this is my first paper about plants. Obviously, it's nothing original. I don't have the knowledge to describe new species of plants, but with my friend Octavio Arango and his help, I wrote this paper about the vejeques in La Palma. And this has a very nice aspect that I was able to accompany him to La Palma for of his expeditions, because a few years earlier, I had seen, when I was writing this book about biodiversity, that somebody had described a new species of plant in the Anaga Peninsula. And if you're familiar with Tenerife, it's like a triangle, like a triangle like this. And this part is Anaga. And Anaga is where everybody in Santa Cruz And in La Laguna go for picnics, they go running, they go just to walk. So it's a very, very well-known place. And how could somebody in 2022 describe a new species of plant in this place? So I was intrigued, and I asked him to accompany him to La Palma. And so we went there, and one of the days we went to Cumbre Vieja. That's where the latest eruption took place in 2021. But Cumbre Vieja is a long series of craters and volcanoes. It's a very nice place. As you can see, it's a bit mysterious. And we reached this crater that is full of these little plants here. And these belong to different species. One is this one, it's called Aeonium spatulatum. And the other one, I believe I have, oh, well, not yet. We have here Octavio risking his life to collect a particular specimen. And this is the other plant, Grenovia ignea. He described this species, I think 3 years ago, something like that. So these two species grow together here, isolated from the rest of the world. And his theory is that if these two species from these close genera live together and they bloom at the same time, they will be hybrids for sure. So his task here was to find hybrids with flowers, because without the flowers, he could not describe them. And this is the plant again, the Grenovia ignea. You can see these beautiful colors. And this is the hybrid. In this case, he found some hybrids with plants, and he described the hybrid, and he was generous enough to give this hybrid an interesting name. So now I'm happy, I can die. Okay, then, and the other part is the limits of life. I have always thought that what in genetics are mutants that are used to understand the wild type, in ecology could be done with extreme environments that are kind of mutant environments. And I'm going to show you two little stories about this. One, you've heard already apart because Bea presented a lot of her work on thermal springs, but I'm going to show you more than the serious part, the fun part of this. This work was done by Bea and by her former student, Estrella Alcamán, in Chile, and by Lorena Ribe in Costa Rica. And the kind of place that we visited is this one. You will agree it's a magical place. It's remote in the Valdivian rainforest of southern Chile. And we were, I mean, you will think that we were very, very lucky to be sampling this place. But I'll show you how to get there. This is a fjord. This is the river. And here's the thermal spring. I've shown here only 3 places where you have to cross the river. There are actually 6. And then here, the river is so narrow that you have to climb with a rope and climb back down with another rope. And this is, of course, it's a beautiful place. This, as I said, this Valdivian rainforest is wonderful. But you have to, well, handle these kind of bridges, and then you have to cross the rivers. Here you have Bea crossing one of the rivers. In this case, there was a rope, but in other cases, there is no rope. And then the guy, this guy, Boris, he was wearing a nice wetsuit, so he didn't worry about the water, but our wetsuits were full of holes. So we were drenched with the cold water, and then the stream was really handy. The other thing is he was like a ballet dancer. He jumped from one rock to the other and then turned around. And in a second, he was on the other side of the river. But of course, we didn't have his abilities, so we had to suffer crossing the river and hanging there as we could. As I said, there were 6 crossings, and sometimes you had to hang to the world. This is sold as adventure tourism, which is fine. But if you're carrying a backpack with instruments and samples, it's not so fun. And here you have the ropes. This is Estrella in the upper part. We have another lady on the lower part. So for some reason, I was surprised because for me it was easy. It's a rope with knots, and you go up, and it was easy. But all the ladies said this was the worst part. For me, the worst part was crossing the river. But so this is a difference with X and Y chromosomes, I guess. But of course, the nice thing about this difficult trip is at the end, you can relax. and enjoy yourself. Okay, then we have different thermal springs. This is Winai, the one I showed you, but we also have the springs in El Tatio that Bea showed you, in Costa Rica, and in Antarctica. And in each one of these places, we had several thermal springs at different distances. So we had a whole set of places, and we could use these as islands, hot islands in a cold desert. And we wanted to know whether the bacteria were able to disperse from one to the other, at least how far they could do it. So here you have a classical distance decay curve. In these curves, you have here the distance between two samples. For example, this point here shows two samples which are very far away from each other. And in this axis, you have the Bray-Curtis similarity. So these dots here are very close in space and very close in community composition. These ones here are very far away from each other and very different in composition. And as you can see, there is a decay with distance, which is what you would expect from ecological theory. However, look at these points here. These are very far away in space, but they're very close in composition. So this deserves some kind of explanation. Here the samples are put in an MDS diagram, and you will notice that the first axis is temperature. We have here 70, 59, 40, 42, 33. So this axis shows temperature. But the second axis, the vertical one, nicely separates the thermal springs from Central America from those from South America. So there's a difference between the two hemispheres, which is kind of intriguing. But let's look at the composition of these different samples. In these more warm ones and the hot ones, most of the biomass is formed by this one OTU, Chloroflexus. And this Chloroflexus is the same one in this square, blue square, which is El Tatio, and in this asterisk, which is in Costa Rica. So at the highest temperatures, there is no problem for dispersal, and the same bacteria are dominant in the different places. So as I said, this demonstrates that dispersal is possible. However, when conditions are not so extreme and the temperature goes down, then competition with the local bacteria it makes it impossible for these bacteria to colonize them. So I'm sure they're there, but they cannot grow. And I thought this was a nice way of looking at dispersal for microorganisms. And then the second example is from northern Chile, from Salar de Atacama. And here the work was done by Cecilia de Mergasso, who is the PI. Camila Scuti, she's the magician with qPCR. She's fantastic. Then Priscila Bendaño did the culture work, and Roberto Veliz was the bioinformatician. Again, the environment is, the environment is very nice, very spectacular, and it's like Bea said, it's a dream for a microbial ecologist. Then what we wanted to know was how water activity affected microorganisms. Water activity, I'll explain, is this. You have pure water. There is a partial pressure of water in the atmosphere, and this is P0. You have, for example, a tomato. There is a partial pressure of water, and here you have a P value. If you divide P by P0, you have the activity of water. So the activity of pure water is 1, and if there is no water, the activity is 0. Now, the current thinking is that below 0.5, 0.6 of water activity, life cannot exist. So we have the system in Salar de Atacama, and you'll see here some rectangular structures that if you look in more detail, well, they're obviously human-made. These are salt urns that are used to concentrate lithium. As you know, lithium is more expensive than gold, maybe, and is something for the future, so it's understandable that they have these very, very expensive solar salt urns. The procedure is to pump the groundwater to the surface, which is already salty, and then put it in these ponds and to let it evaporate. Since this is 2,500 meters above the ocean, it's very dry. Solar radiation is very intense because it is in the tropics, and there's wind. So this makes evaporation feasible. As the brine concentrates, it changes color, and eventually gets to this cough syrup yellow color, which is really offensive because of the vapors of chlorine. And what happens along this gradient is that, so here is the beginning of the groundwater, here's the lithium concentrated brine, is that different salts precipitate as they reach the solubility product. And some of them are used, for example, a potassium chloride is used for other purposes. The other thing that happens is that lithium increases 10 times, so it becomes commercially feasible to exploit it. And the other thing that happens is the salinity goes from about 10 times the seawater salinity to about 20 times. So, of course, along this gradient, you have here the lithium concentration, and here you have the activity of water, and the activity of water goes down, while the temperature goes up. And if you look at the values of this water activity, 0.5, 0.6 is here. So in theory, there can be no life beyond this point. And our idea was, is there no life? Let's check. So you collect samples. As you can see, these workers have to protect themselves from the tropical sun and from the vapors of the chlorine, so this is not simple. Well, we rinse the containers and we collected the water. We took it to the lab. And then what you usually do is that you filtrate the sample, you extract the DNA, and then you sequence or do qPCR. The problem is that this brine is extremely viscous. It's offensive, it's disgusting. And if you try to filter, only one male or maybe two males go through the filter. This is not enough to have good DNA. And the result is that you have only a few males, the poor quality of the DNA, and the bad sequences. So we had to do something to avoid this. And what we did was to introduce a step of dialysis. The idea is that we put the brine inside the dialysis bag, we put it in water, distilled or deionized water, we let this equilibrate, and hopefully the ions of salt will go out, but the bacteria will remain in, so we will be able to filter and sterilize. Of course, we were concerned that maybe the bacteria would blast, like do the archaea in solar salters, so we had to fix them before. And so we had to fix with 50% ethanol. Now you can imagine what it is to work with 5 liters of ethanol and 5 liters of this disgusting brine. It's really a very tough job that Camila and Priscilla really fought for many months. And at any rate, this is the way it looks like. You have here the dialysis bags, and controlling the conductivity on the outside, you know when you reach the point of equilibrium, and then you can get your samples and sequence. So you have these 3 elements, and we wanted to make sure that there was no contamination. So the first thing we checked was that the reagents for the qPCR were not contaminated. When you do a PCR, there's always a sequence coming. This happens all the time. But if we found in our samples a sequence identical to one of these, we would discard it. The same thing with the ionized water. This water doesn't get in contact with the sample because there is the dialysis bag. But even so, if we found a sequence identical to one of these, we also discarded them. So with this procedure, we discarded, I think, was 2% of all the sequences. So we were quite happy. And then we had to have a positive control. That was adding a known culture fixed with ethanol and then recovering it appropriately. So we did all the controls we could think of. And then with this methodology, this is what we found. What we have here, this is the water activity. Here is the value, so here is 0.5, and it goes down. And this is the abundance of bacteria in blue and archaea in orange. So you can see that we could find bacteria in most places. There was something that didn't work here, but we could only find archaea at the beginning. So this was shocking because in regular solar salters, the archaea increased their importance in the saltier ponds. And this was not the case here. They disappeared. And the bacteria, on the other hand, were able to be here. Now, look at these values. In reality, this is 10 to the 2. So this means we have about 100 cells per milliliter, which is very little. There are some groundwaters that are like this, but it's very little. So you can think, okay, we have bacteria, but they have been pickled with the salt. So they're staying there, and they're not doing anything. So in order to see whether this was the case, we looked at the ribosomal RNA, not the DNA, but the ribosomal RNA. And this is what we found. The purple is again the same as here, is the same abundance. And the green is the activity. And as you can see, we saw activity in the lower salinity ponds, which is what you would expect. We didn't see it here, which is what you would expect, but we have some here. And look at this activity of water. It's about 0.3. And in this case, it's about 0.5. So these are at the limit, at the no limit for life. And this is beyond that limit. This is not possible. Life cannot live at this low water activity. And yet we find ribosomal RNA. Notice that we don't have RNA for archaea, so they were not active in any place. So you could still say, okay, the RNA under these conditions is more stable than under normal conditions, and maybe this RNA is not showing activity. So let's try to grow the bacteria in culture. And this is what Priscilla did. She got the water. In the low salinity ponds, she would filter, and then the filter had the cells, and the filtrate had the sterile brine that we would use as medium. In the cases, the more concentrated ones, since we could not filter, she would centrifuge, and again, she had the pellet with the cells and the sterile supernatant. So this was the medium we were going to use. She added LV, metals and vitamins, she enriched them, and then she added to the experimental flasks. To the experimental flask, she added a filter, and to the control, she didn't add anything. And I will magnify this slide. This is the control after 10 days, transparent. These are the experimental after 10 days, turbid. So the bacteria had grown here. And she looked under the microscope and she could actually see cells. Again, this is impossible. How come that they can grow at this very low water activity? It is true that when we add the LB medium, we're sort of reducing a bit, increasing a bit the water activity. We're adding some water. It's possible that the medium is not homogeneous, and there are some microhabitats where they can have a little more water. It is true that some of them had capsules, and maybe these polysaccharides retain a little bit of water. But still, we don't understand this. And the problem is that we're about to send the paper to a journal, and we know what they're going to say. This is not possible. This is contamination. So we need your help. Okay, to finish, I'll talk about my last enlightenments. I was here in southern Chile, in Huinay. It's a national park. It's the closest landscape I've seen to paradise, with the volcanoes and the lakes and the Araucaria forest. This is shown in every dinosaur movie. They always show this landscape. And it's full of it has a few little cabins distributed in the forest. And we were staying with some friends in one of these cabins, and they're far away from each other. So you have the feeling of being alone. We were having our pisco sour and discussing about everything human and divine, our past lives, our expectations for the future. And of course, night came by, and maybe after another pisco sour, I have to confess, I looked up to the sky, and I could see the stars, the Southern Cross, and the constellations of the Southern Hemisphere. And I could see that a little bit further down, well, a lot further down, we had the canopy of the Araucarias. And then on the floor, it was us. And I realized that the stars have life cycles of thousands of millions of years. The Araucarias have life cycles of thousands of years, and we have life cycles of a few decades. So again, these magnitudes of 10 of different structures embedded one within the other and working at different speeds. And I thought this makes a wonderful world. And I thought that science is a wonderful tool to study this universe. So here's my advice. Don't ever let anybody take away from you the pleasure of doing science. Thank you.
doctor, get flowers.
Okay, so I think thank you very much to everybody that has been here. I remember that there is food because it's quite late, and yeah, that was a very nice morning. Yeah, anybody that wants to just questions, Carlos will be there. We are going to be there. So.
Thanks. Sí, thanks, Carlos. This is so different, Pep and Carlos. Hola? 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 El Pep, com sempre, no s'havia donat compte que la Cèlia havia aixecat la mà. I cannot say. So I wanted to say thank you. Thank you in big, big letters. But you say something very important here, that you were doing all this because you have curiosity and you want to have fun, which is the last message. Actually, you start with this and you end with this. And we can all see that. You didn't this to have power. You didn't see to be a leader. But you have the power of transmitting science so nicely that we are here for this. And you don't know, but you are a leader, although you don't want to be a leader. So thank you so much to have these big examples for us and for the next generation. This is one. The second one is that you say, because you are another quality of you are very modest. You say, I didn't give ideas, were your ideas. Maybe, but my experience, and I think most of our experience, is that when we were talking to each other, you always have a point of view that was from other angle, other sphere. So this gives you the opportunity to enter in a world of new ideas and new questions. And maybe it's not one idea, it's a lot of ideas that came from these different angles of. So thank you for. No. Thank you for that too, but this is another thing that I want to highlight, very, very highlight, is that you are the only few scientists I know that you give credit to the woman when it's necessary. You encourage women to do science. You encourage women to be independent. You encourage this to everybody, but I think women appreciate this very much. Thank you, Carlos, because I feel all these things on my career together with you.
Foto aquí.